Good morning, everybody. We're going to make a start, so if you want to take your seats, that would be great. Everybody come through and sit down. There's plenty of seats. Dr. Griggs, you can sit down if you like. Okay. Great. So welcome, everybody. It's really fantastic to see you all here at this fantastic venue, which is actually amazing and quite historical, as you all know. It's great to see colleagues from across America, from Europe and internationally, and also to welcome our industry partners who are a key part of this meeting. This is actually the 25th meeting, which is quite amazing, really, and obviously the whole story was started with Birch Griggs, who I'm delighted is here as well today. We think we've put together, thanks to the planning committee, a really interesting and varied program which reflects the amazing progress that is happening in neuromuscular diseases and the real potential now of therapies for many of our patients, which is really crucial. So we really hope you have a fantastic 2 days of networking, of science, of clinical practice, of trials, and learning the latest things that are happening in our field. But first of all, we thought it would be interesting just to look back a little bit to the history of the NMSG, or MSG as it was. And of course, Rick is an aficionado of all types of history, so he's going to talk you through.
We're going to do it together. Don't move. So good morning, everyone. Good morning. I'm so great to see everyone here and to reconnect with old friends and to make some new friends. So this is, as Michael said, the 25th anniversary of the NMSG, if you count it the way we are counting it. So I think we're right. I think it's the 25th. And so we've done a lot of neuromuscular archaeology over the last few weeks to try to get this timeline together. It's been a combination of my team, the team in Rochester, trying to put these facts together. So this is the way I think it started. I think it started, Dr. Griggs, in 99 when you called a few of us to a meeting at the Chicago Airport Hotel. And you had this idea to start this group. Tony's nodding, Birch is nodding. There's not many people left from that original group. So. And then we had, I think, our first inaugural meeting in Rochester, is my recollection, in 99. So we are saying 99 is the start year, which makes this the 25th anniversary. We could be wrong on this, by the way. So I'm just saying, that's the story, and we're sticking with it for now. And then we quickly pivoted, and in 2001, we had our first meeting in the luxurious Beaver Hollow accommodations in upstate New York, outside of Buffalo, which I see Dr. Howard smiling about Beaver Hollow. It was lovely. It was really great. And some people are telling me that this place reminds them of Beaver Hollow, and I'm thinking, how? So we had a number of good years at Beaver Hollow, and then Dr. Hanna joined the group. When do you think you joined? 2005 or 6? Yeah, we think Mike Hanna joined the group in 2005 or 6, and he convinced Dr. Griggs to move the meeting to Oxford. And so we did. Yeah, right. We got an Oxford slide here in a minute, but it's coming up. So we did Oxford, we went back to Beaver Hollow, and then in 2014, for reasons I still don't understand, Dr. Griggs said, I'm done leading this group. Mike and Rick, it's your turn. And so we actually had an election of sorts. And Mike and I were elected to be the chair and the co-chair of this great group, which we're still in those positions. And that was in 2015. Well, actually, the election was in 2014, right at that last Beaver Hollow meeting. And so then when we took over, we moved the meeting to Snowbird, Utah, because of Gordon Smith, and a lot of our colleagues at the time were in Utah, and they told us about this great conference place, and it was great. Snowbird was also sort of a magical convention center. You really couldn't get away, you were trapped, so we all had to, do a lot of work together. That was a lot of fun. And so we had many years in Snowbird. Actually, the other big thing that happened in 2015, when Mike and I took over, we made it an official nonprofit 501c3. And before that, Dr. Griggs ran the organization through various bank accounts. I really don't want to know how he did it, but we became legal in 2015. And then, so after those Utah years, COVID hit. So we had 2 Zoom meetings during the COVID years, which were hugely attended. I'll show you the numbers in just a minute. And they were very successful. And then when we were emerging from COVID, Valeria Sansone, who's one of the original members of the group, came on about the time Michael did. She had this great idea to do the meeting in northern Italy. And so we were in Stresa, Italy, which was truly a magical place to visit. And then last year, we went to Orlando, which I think was my idea, so I apologize. I mean, it was a great meeting. It really was a good meeting, but it was Orlando. So now we are here in Tarrytown. And why are we here? The executive committee gets together multiple times a year, and we're always brainstorming where to do the next meeting. And we give Liz Polk ideas, and she runs them down, and some places are way too expensive. I don't know why I forgot about this place, but over 20 years ago, and Jackie Montez may be able to tell me the year it happened, but Hiroshi Mitsumoto had an ALS meeting here at this conference center that I was at. Jackie, where are you? What year was that? 2004? It was in the early 2000s. And I'd forgotten about this place. And so when we were brainstorming, somehow it rose to, the idea rose back in my brain, and we checked it out, and it's still here. And it's a great convention center. So that's why we're here in Tarrytown, is because of Dr. Mitsumoto. So let him know that, Jackie. And then next year, by popular demand, we're going back to Italy. Dr. Sansone is setting that up. Okay, next slide. That's my history of the NMSG slide. Oh, there is. Okay, I'll wait. I'll wait till I show the Viva Hollow pictures.
You go back one.
Did I mess it up?
Go back one.
Oh, you're right. You're right. Okay, so for the first time, we have metrics of this conference, and Liz Polk and Amanda Seebach and a lot of people on the team just did an amazing job doing this sort of NMSG archaeology. And this is the abstracts, the number of abstracts that were presented at each year since, I guess, the beginning, Liz? Almost the beginning. This is not, this is attendees. I'm sorry, thank you, Liz. This is the attendees. But since pretty much the beginning, and you can see the number of people coming to this conference has just gotten bigger and bigger. And so there's this blip for the COVID year. But it's still, if you sort of take those 2 dots out, you can see we're continuing to increase. And this year we are up to 270 people. And then we have some timelines there. 2008, first year we had the abstracts published in Muscle and Nerve, and then in 2020 was the first year we moved to the RNMF journal. All right, Michael, you're next.
So the top red line is really just the abstracts submitted, and that's obviously continually increased. The first spike is COVID, but the second spike, which I think is quite interesting, is when we go to Europe. So that's the Italian spike. So that's all good. And then the lower green line is just a reflection of the number of companies and pharma colleagues attending, and this has just shown a progressive increase, which just reflects where our field is at, which I think is very, very positive, and we are really happy to work with them as partners.
Which leads you to this slide.
So what's the NMSG all about? I think that a key thing for all of us is to support and develop young investigators in the field. I think it's really true that although the last 20 years have been pretty exciting, the next 10 or 20 years are going to be amazing, because I think there are disease-modifying therapies across the genetic and acquired neuromuscular diseases that are really going to make a difference. And so it's a really good time, I think, to be a young investigator going into this field, because you'll be the people who will be deciding and managing all of these different conditions in a very, hopefully, effective way for patients. So I think a core aim of the NMSG is to support all young investigators across the world, actually, who are moving into this area.
Hey, can I do something real quick?
Yeah.
Inaudible?
Yeah.
How many people in the room consider themselves young investigators? How many people want to be a young investigator? Okay, all right, there you go.
Yeah. Yeah, but we have a good number, and that's really great to see. So fostering international collaboration is key. It's key for trials, but it's also key for developing networks, which really is important, and I think is something that I benefited from as a young investigator when Birch first invited me to join. And I think this is an American circle. We want to be the premier organization and annual meeting for neuromuscular clinical trials in particular, and there's lots of trials that we have run through the NMSG. We want to connect clinical investigators to preclinical colleagues, so we can see what's coming through, and making that connection is really important, and that's been something I've done in my area where we've connected with work Steve Cannon's done, and then taken stuff into periodic paralysis patients, for example. And of course, as we've said already, engaging with colleagues in industry is increasingly important, and it's great to have such interest in our meeting and our diseases from our industry colleagues. So those are what we think are the key goals.
Okay, now here's some great pictures. So those of you who have been around a while will be able to find yourselves in these pictures. These we got from your Rochester team, Birch, and these are the Beaver Hollow pictures. I love Beaver Hollow. One of the things that was interesting about Beaver Hollow was sometimes you didn't get your luggage in time. It got lost at the airport. So one year, my luggage didn't make it, and so I had to get an emergency wardrobe, and there was no Target or anything. But Beaver Hollow had also become the base camp for the Biggest Losers weight loss program. So I had to have a whole outfit of Biggest Loser clothes for at least 2 days of the meeting. I don't know if people remember that. And me and Maureen Walsh and a couple others were wearing Biggest Loser sweatsuits. I still have mine. I don't know what happened to that group, but one of the reasons we had to move out of Beaver Hollow was not only because of the Biggest Losers, who were taking up more of the rooms, but the conference site just got too small for us. This group really grew. I don't remember how many we were able to accommodate there, 150, Birch? Something like that. And the conference just kept growing, and so we outgrew the Biggest Losers in Beaver Hollow. And then we went to your part of the world.
So after 14 years of it only being in America, we moved to Europe, which I think was a significant development. But I'm biased, obviously. And in 2013, we went to Oxford for the first time, and then we repeated that in 2018. I think they were very successful, very well-attended meetings. The main concern was the rooms were much smaller than colleagues were used to.
Very small.
They were student dorms,
small.
but they were very successful meetings, and we hope to take it back there again.
We'll go back to Oxford one day.
Yeah.
Maybe bigger rooms.
Yeah.
And then we had the Snowbird years, which were a lot of fun, and thank you for our Utah colleagues. Many of them are here. Some are still at Utah. Some have moved on to other parts of the country. But that was a great suggestion by Gordon and his team to have this conference in Utah. we got a variety of these pictures from various places. The one on the bottom left, your left, was the year we had a blizzard in September. I don't know how many of you remember that. And we were literally snowed in for 3 days. So we had to do the picture inside. But most years it was spring, fall time. It was lovely. All right, what's next?
So then, of course, there were COVID years when we went online, 2020, 2021. Incredibly well attended, as was observed across online meetings, but clearly less good for networking and linking with colleagues, which is so important. But we kept the meeting going during those years and had really high attendance. So that's that one.
Which makes you wonder, the attendance was so high. We talked about a hybrid meeting, but we just never did it. And I don't know, we can talk about that. Tony's shaking his head. Okay. We won't do that, Tony. Then we went to Italy, and look, there was that beautiful, beautiful hotel conference center, which everyone loves so much. We're going back next year. And then Orlando, which is great. It was a great meeting last year.
So this next
All
slide
right, this
is Rick.
slide we were advised not to show. One of my assistants who's been with me for many years, Amanda, said, Dr. Barron, you do not want to show this slide. So, but we are. So NMSG is about advancing scientific knowledge to help our patients and their families with their neuromuscular problems. That's the main goal of the NMSG. But we also, in the evenings, have fun. And so this is some pictures of that. I think it was in Beaver Hollow because it was outdoor. And every night at Beaver Hollow, the place put together a campfire with marshmallows and s'mores. So I'm blaming it on Beaver Hollow. But we had a lot of nice campfires with fun. And then when we went to Zoom, we continued to have our Gil, our AV guru, set up a cigar room on Zoom. And Vino, you're there. Just saying. And our sisters, where are you? They're up in the top right. Yes. So your crew, Michael, was very active in those events.
Yeah.
I'm just saying.
Yeah. Next slide.
All right. T-shirts have been a big deal for the NMSG, and this was all started by Dr. Griggs and his team. They always gave us nice T-shirts, and so the top line are from those Beaver Hollow meetings. And then the Beaver moved to Oxford. So in 2013, we cannot find a 2018 Oxford T-shirt. If anyone has one, take a picture of it and send it to Liz and me. We were back in Beaver Hollow, and then when we moved to Snowbird, the Beaver had to learn how to ski. So you could see the Beaver on skis. Then the Beaver got European citizenship, and then was playing with Mickey Mouse. And this year, the Beaver is headless. Why is the Beaver headless? All right. So why is the Beaver headless? So for those history buffs, this is the hometown of Washington Irving, and I've been a big Washington Irving fan for a long time. And here's one of my copies of, this is printed in 1901, and he actually, what happened to our slides? He actually did these, he wrote in the early 1800s. He was born right after the Revolutionary War, and he was the real first man of letters in the United States of America. Him and Nathaniel Hawthorne, I guess. But he was most famous for his two stories, Rip Van Winkle, which took place in this part of the world, where he said it, and then, of course, The Legend of Sleepy Hollow, which was a German fighting for the Americans who got his head blown off in the American Revolutionary War, and his ghost would haunt Sleepy Hollow. And so Ichabod Crane, who was a minister and school teacher, he got into it with the ghost one night, and he disappeared. So that's the legend of Sleepy Hollow. And I found his house down the road. It's about a mile down the road, Washington Irving's house, and I'm going to try to get there. It was closed yesterday when I went. All right, enough history. Dr.
So yeah, just to shout out and thank sincerely all of the planning committee who put together the program for this year's meeting. The least important people on that list and the ones who did the least work, me and Rick at the bottom. Everyone else really worked hard and put this program together, and we're really, really grateful because it's extra work. We recognize that. But your networks and your knowledge has allowed us to create a really varied and interesting program. So I think we should give a round of applause to the program committee.
And I got to add that while Dr. Hanna's and my name are at the bottom, we actually did nothing to get this meeting. Yeah, so it was all the committee. Thank you. Yeah. And our executive committee?
So yeah, again, this is just the colleagues on the executive committee, just to thank them all for their work over this last year. And just again to recognize that Birch really set the whole thing going, and he is chairman emeritus forever, which is great, and we're really grateful to Birch for everything he's done. But all of the members of this committee provide really valuable knowledge and insight into steering the whole of the NMSG as we move forward. So thank you to all of them as well. And then we want to welcome incoming executive committee members listed here. It's great to have new people rotating in, and we're really pleased to have these colleagues joining us and look forward to working closely with them.
People on the executive committee, can you stand up so everyone knows who's been involved? Thank you. All right, good. Thank you very much. And then does that include then Amanda, Basker, and Michaela? Did you just stand up too? If not, just stand up. Yeah. All right. Thank you for doing this. Almost done.
Almost there. So just to sincerely thank all of our sponsors for helping us make this meeting happen. We have an interesting industry session as part of the program where science that they're doing is presented. So these are our platinum sponsors, so thank you to all of them. These are the silver level sponsors and also the bronze level sponsors. So the meeting could not happen without them, and you're going to thank Liz as well.
Oh, that's the next slide, but you know, just on the sponsors, this meeting has now become, it's really changed over the last 25 years, and it really has become a meeting of industry representatives and neuromuscular frontline health professionals and scientists. So we want to make sure that all of the healthcare professionals and industry folks do a lot of talking over the next 3 days, not only go to their booths, which most of them are in front here, but some are in the front when you walk in the mansion, but at all the social sessions too. Make sure that you get to know each other so we can come up with new ideas to make new neuromuscular discoveries. And then this is the people who we really need to thank. And let me just go through them. Liz Paulk. Liz is standing in the back there. Everyone knows Liz Paulk. She's I probably don't have to tell you how hard she works all year long, and then in the months coming up to this, it's a lot of work. So thank you, Liz. Gil in the back has been with us now, our AV guy, for a number of years. 4 years. Where did we find you, Gil? We found you in Kansas. You actually helped us in Kansas City for an event, I think. We found you in COVID. Oh, that's right. We Googled you in COVID, and we found you. And he's been part of the team ever since. That's right. Thank you. And then, where's Laura Herblin? Where are you, Laura? Did she step out? Laura tried to retire. She's doing yeah. So Laura tried to retire, but she's back working part-time. She's and helping us a lot on this meeting. We have a medical student here, LaDon Bigdelli. Where are you, LaDon? She's in the back. She's an Ohio State medical student. She's next to Gil. She's been working with us for 2 years now. I think Dr. Arnold found LaDon for us when he was at Ohio State, and she's amazing, even though she's going into ophthalmology, but that's okay. And then several people who aren't here are folks that are at the University of Kansas, where I am still an emeritus professor, and so Grace Reep has helped in the design department, and then Marion Reed and Eric Bader are in charge of the digital press operation that allows us to do the journal. So what's happening today is, as soon as Mike and I get off the stage, we're going to have presentations of selected abstracts, platform presentations. We're going to have some selected flash presentations. We'll have the new and young investigator, evaluator, and coordinator session. And then tonight there's a poster session in the carriage house. And then there's a dinner and reception tonight. Just to remind you, our abstracts have been published last week in the newest issue of the RNMF Neuromuscular Journal. This is the link to the journal and the DOI of the abstracts for your CVs. And just a shameless plug for the journal, I'm trying to keep this journal alive. It's really a labor of love and ego. So I don't want it to die. So please submit articles, many of you have, and particularly abstracts from this meeting. If you want to, you can assure they will most likely get accepted. I can't promise, but most likely you can get your articles published in this journal.
So then just a few final housekeeping points as we start the sessions. The entire meeting is being recorded. After a presentation, if you want to ask a question, raise your hand and we'll bring a microphone to you. The Wi-Fi access code is there. There are charging stations in this room, and obviously coffee and water and snacks in the room behind. So I hope you all have a really useful, interesting, and enjoyable meeting. We're going to hand over now for the first session on genetics to Vino and Carolyn. Thank you.
Okay, we're going to crack straight on. So our first session today is on genetics, and this is a very exciting session. Genetics really has revolutionized neuromuscular disorders, both discovery and treatment. So we're very excited today to introduce our first speaker, Emma Cefaloni, and she'll be speaking on gene therapy for DMD and SMA, milestones, lessons learned, and current challenges. Thank you, Emma.
Thank you very much to the organizer. I'm very excited to be here and to talk briefly about something that has been so revolutionary, changing our field, and so very rewarding. These are my disclosure. I'm going to try to give you the 30,000-foot view of some of the big lessons learned, not just from the science, but also from implementing gene therapy into clinical setting, and how much we can translate from what we learn for spinal muscular atrophy into Duchenne, and yet how much difference there is really when we look at the challenges, difference between SMA and Duchenne, and how much harder it is to actually target a tissue such as skeletal muscle. And as you know very well, more than 50 years of science and clinical trials for gene therapy, some of the most fundamental lessons learned after the hyper-optimism of the conclusion of the Human Genome Project that was completed in 2003, and the hyper-optimism that gene therapy will be around the corner. One of the very important lessons was learned, unfortunately, from the death of Jesse Gellinger at the University of Pennsylvania that died in 4 days after a fulminant immune reaction to the vector. And the two fundamental lessons there was one, that really, in order to translate gene therapy safely, we will have to learn how to deal with the immune reaction to the vector, and later on, also we have learned about the transgene. But the other lesson was also that the way gene therapy are carried out really needed a lot of redo. Many problems were found, for example, in the way the informed consent was obtained, and so there was a big halt in the majority of gene therapy trials because of that case. The other big lesson was insertional mutagenesis, with the first success of gene therapy in SCID that was performed in Europe with kids that really were successfully treated and with quite an impressive durability of the gene that was transduced. It turned out that 5 of those 20 kids actually developed leukemia due to insertional mutagenesis, and that was also a big lesson that really helped to move the field towards avoiding that. But then after this Downs moment, really the field has taken off, and is really the exponential speed of FDA approval in the past 5 years or so is quite remarkable, with 36 FDA approved gene therapy as of March of 2024. And it's also very remarkable to think that although the large majority of clinical trials in the last 50 years were really targeted at cancer diseases, and they were mostly based on ex vivo transduction, like CAR T cells and many other, really, we in pediatric neuromuscular took the lead with the first in vivo transduction for SMA. And so many of the lessons learned there really came to be learned in pediatric neuromuscular setting. And some of the milestones, only in 2023, there were 7 gene therapy approved, and December 2023 was called by many the most exciting day in the history of gene therapy, where on the same day, 2 gene therapy for sickle cell anemia were approved, one of which is the first CRISPR-based gene therapy. And the prediction in the field is that from 2025 on, we will probably be looking at 10 to 20 new gene therapy approval each year. So one of the major milestones, certainly for our neuromuscular monogenic diseases, is what we have learned about recombinant AAV, and that these really have many, many good advantages for our application. They have high transduction efficiency, they have no known human pathology, and really the ability with very different serotypes to target different tissues, and they have different tropism. Overall, they have a low pre-existing immunity, although when you look at the epidemiology globally, this will remain to be seen. And really, they're very well set to be used for systemic delivery. And importantly, they're non-integrating, so really avoiding the risk of insertional mutagenesis. The disadvantages are that indeed patients, different ages, do have pre-existing antibody to the wild type. So although this is not a major problem in infants with SMA, because those are transplacental and they clear fairly rapidly, in a disease where we treat older children, like in Duchenne, then becomes more of a barrier. There is 100% seroconversion after dosing, so difficult to imagine redosing. And they do have a small payload capacity, which not a big challenge for a gene like the SMN1 gene. obviously a much bigger challenge in larger gene like in Duchenne. And then for skeletal muscle, a very, very high dose is required to reach the target tissue. So SMA, when we have all heard the story and we know the data, but really there were so many perfect, beautiful storms that allow us for this to be so successful, one of which is the AAV9 is able to deliver across the blood-brain barrier, so to reach the anterior horn. The vector is able to actually accommodate the full length of the SM1 gene. Again, designed not to integrate into the genome, so it really sits in the nucleus as an episome. And these are non-replicating cells, which also extremely helpful. And then the CB promoter really allow for really fast onset. And having seen this in clinic, is incredible how this baby, once you treat them, you really see them back in a week and 2 weeks, and their CHOP is going up, which is really quite an incredible thing to witness. The other perfect storm, and I think that this group, we have learned from Birch and many others, the importance of strong natural history data. And I think that SMA is the most brilliant lesson, that when you have a well-defined natural history, these are the data that we know that, 8% free survival at 20 months for SMA type 1. When you have that well-documented natural history, you can actually carry out trials. And this is the first 15 kids treated in SMA, where the primary outcome was really ventilation-free survival, and you can see that all 15 survive well past 20 months. This is very, very hard to argue about, is this a positive trial or not. One of the major milestones is really that have revolutionized the way we really diagnose rare diseases is the success of SMA with now 3 FDA-approved drugs, including the gene transfer drug, is that as of January of 2024, we now have 100% newborn screening in the USA. It took about 6 years from the time the RASP actually recommended to add SMA to the universal panel to actually implementing it in all 50 states. And this is really transformed the way we treat, because as we have learned from subsequent trials, when babies are treated very early, before 6 weeks of age, this is the phase 3 SPRINT trial for babies with 3 copies, you really have a chance to treat early, and you can see in the 2 panel, that's the Bailey scale, and for gross motor and fine motor, these are babies that develop exactly along the curve of normally developing children. So the gray shade area, that's normally developing children, and that curve goes up to 24 months. But when you look at babies with 2 copies, so destined to develop type 1 SMA, and this also is the phase 3 SPRINT trial, although the rapid improvement, it's incredible, and you can see the CHOP intent on the top, so they all improve. You can see that over time, even when you treat at the pre-symptomatic level before 6 weeks of age, you can see that the Bailey in these babies, there is clearly a subgroup that falls below the normally developing curve of babies. So really telling us that this, even in the best implementation setting, is not a cure. And this is an example of a 2 copies baby that was treated at 14 days. We have improved our ability to treat, and I think in our center, we've treated somebody at 7 days. So obviously, these are milestones that for a 2 copy babies are unthinkable and completely excellent. But as you can tell, these are very delayed and still clearly outside of a normal developing child. So, and this really opened the question about add-on therapy and combination therapy for this group of 2 copies. And really what SMA has taught us is that really has changed everything that we do in terms of infrastructure. So from an old model of diagnostician and supportive care to really a new model where we are called from health department. We are actually the base clinic for newborn screening. This is really time is muscle. You have to really have a 24/7 team ready to move on these newborn screening positive cases. And really having the infrastructure in place really is so hugely important to really change the outcome, because the sooner you treat, the better the outcome. And what it really takes, what it takes to really have better outcomes, and we have learned that in the early days, we thought that, treat before 6 months, treat before 6 weeks, and now we're at 7 days. And we have seen babies that you are really looking at them at day 3 or 4 or 6 after birth, and they have good CMAPs, good reflexes, excellent CHOP, and you see them a week later and they're now lost reflexes and clearly become symptomatic. So what it really takes is many of these practical access infrastructure that really include many different people that I didn't even really know existed at my institution, because as a pediatric neuromuscular, all we had for the longest was prednisone. But now we really have to advocate and be a champion to really bring your finance team and leadership and pharmacy, and really to create an infrastructure that goes well beyond neuromuscular specialists. You need to have liver specialists, cardiac specialists, because of the profound safety issues. And really, this is what is gonna make a center to be really able to dose 7 days versus 7 weeks, which obviously makes a huge difference. So there remain many challenges and opportunities in SMA. These are just some. So the faster, the better. The diagnosis through newborn screening is really not the end of disparities. It's the beginning. The newborn screening is the big equalizer, right? Now you have a chance that everyone is going to be diagnosed extremely rapidly. But what happens after that is still prone to many inequity, geographical access to really centers that have that kind of infrastructure that can make this dosing of these drugs not only timely, but also very safe. And then combination therapy, which obviously for those group of 2 copies babies is the reality, and is already happening in the clinic, where we add other SMN2 modifier. This is already happening in the clinic with lack of any clinical trials data. So we're really, we're doing things without really strong data. And the other thing that we could do a lot better is really long-term clinical outcome. There is an opportunity here to really learn about the new phenotype of SMN. And unfortunately, while there is very many grant opportunity that support newborn screening pilots, it takes millions of dollars. There is very little mechanism to support long-term newborn screening. And while the epidemiology think of long-term follow-up in newborn screening as, okay, are these kids going to school? Are they tucked in a clinic? Are they alive? I'm talking about really clinical outcome. And when it comes to that, it's really very, very fragmented, and there is no good mechanism to capture that. And then the ethical issues. If you do treat SMA in clinic, you know that you had many interaction with your ethics committee, because lottery and waiting list for treatment. But not only that, what do you do when parents decline such a life-changing treatment? What do you do with that? Or when they want gene therapy, but they don't want standard of care, for example, vaccination. So we have been using the ethics committee a lot more. So moving to Duchenne is really a different set of challenges, but obviously we have learned a lot from SMA. But I think that an incredible milestone in Duchenne is that in June 2023, the FDA approved the first gene therapy for 4 and 5 years old, and that was followed in a year later by an expanded label by the FDA that gave a full traditional approval for all ambulatory children 4 and older, and an accelerated approval for non-ambulatory patients 4 and older. So it's really a very broad label. And there is a big lesson here from FDA that really the FDA made this decision based on the evaluation of 2 double-blind placebo-controlled studies, 2 open-label studies, more than 200 patients treated, but really the decision was based on the totality of the evidence. And I think that the message we can read from this is really that the Office of FDA for Cell-Based and Gene Therapy has sent a strong message. They're willing to look at the totality of the evidence in a quite open and different way, and obviously we don't have time to go into the details. But the exciting part about gene therapy for DMD, that this is just the end of the beginning, is the first big milestone. But if you look, there are at least several programs. The Sarepta has now approval, and they're really working on a phase 3 in older and non-ambulatory patients that is enrolling very well, and it's ongoing. So there is a lot more to be learned about older, heavier patients. Unfortunately, the Pfizer, you have learned, their phase 3 trial failed, and they also had 2 deaths. Both were cardiac death in an older patient and in a very young patient. But there are many other programs. The Solid Biosciences went back to the drawing board and now have a modified capsid that has been engineered and modified to really have a much higher potency for skeletal muscle and less liver toxicity. They're also looking at much improved true full capsid. And I think that the empty to full ratio for this gene therapy product is not really discussed openly enough. And in fact, FDA, I don't think, has any specific requirement about the ratio, although it needs to be disclosed. There is really no level. And I think that really has a lot more with the efficacy of transduction that sometimes we think of. Genenton continuing well to progress in UK and France with 5 children now. Those as the program that is led by Dr. Montoni. And Regens Bio is now enrolling very young as well. It started to enroll 1 to 11. And really all of these programs are slightly different cassettes of transgene and different AAV, but the Regens Bio is the only one that actually has the C-terminus domain in their transgene. And so it remains to be seen how some of these other drugs will perform in the future. And so while we have really learned many milestones that you can see here, how to deal with the immune reaction against the transgene, which we learn by now at this point excluding certain mutation in this CRIM-negative patient. We have learned how to manage and mitigate and even prevent immune reaction against the capsid. What still is the big challenge for targeting skeletal muscle, we're probably going to hear more in the next talks, is really durability and dilution effect, because unlike in SMA, these are dividing cells, and really the therapeutic window, when is too early or too late. So we're not treating babies, we're treating children and older children in Duchenne at this point. We need to find the solution for the pre-existing and the induced immunity. if we are thinking already about redosing, and there are trials now studying plasmapheresis and other strong immune suppression protocols for this issue. And then really the risk-benefit for older, heavier patients that receive really very high doses. So one of the big things that people are working on is improving vector to allow really lower dose, less liver toxicity, and really greater power to target muscle and cardiac tissue, allow redosing, and really dose this patient with positive AAV. And you probably have heard of MyoAAV. We have seen BioSolid re-engineering their AAV9. And then still not quite in the clinical realm, but exosome nanoparticle and other strategies to really minimize the immunogenicity of the vectors. The therapeutic window is a huge question because in muscle, we know that there is very early injury of the cells, of the muscle cells, and that really trigger a downstream series of events that once they are triggered, they are continuing on their own. And so the thinking of, is gene therapy in a 1-year-old too soon, or could it really stabilize the muscle membrane to really have an impactful effect on the downstream effect? On the other hand, gene therapy does not transduce satellite cells, and this is a dividing growing tissue. So those remains a big question with quite not clear answer yet. And the other challenges is really manufacturing scale up because of the numbers of Duchenne prevalent cases is very high. The cost of production, but also the price of the drugs, which really takes a very different type of work to decrease those. I think that we need to learn more about empty to full ratio and really overall improve the cost of new drugs, because at Duchenne, what happened, we also had 2 other drugs approved very recently. So we're really looking at a combination therapy that really adds up. And some policies, we have heard people who say, well, if you want to give gene therapy to do Duchenne, you really have to sign and tell us that you will never use exon skipping drugs, or that you cannot add on some other type of therapy. And so that will become really an access issue. Newborn screening is happening with 3 states already starting, New York, Ohio, and Minnesota. And really, this is due to a persistent delay in age of diagnosis, which has been stuck at around 5 years old for decades. And the big issue that we are facing in clinic is that none of the 30 to 40% children with Duchenne that have very profound CNS phenotypes were really able to participate in the clinical trials. And this is really the 4 and 5 years old and 6-year-olds that we're left to really try with gene therapy now. So, and we talked a little bit about the need for really interdisciplinary and financial infrastructure. I just want to leave you with this picture to think about that. Unlike cystic fibrosis, you can see the number there. These are the number of the Cure SMA and parents and PPMD clinics. And with the numbers of Duchenne that are now on waiting lists for gene therapy and other treatment, I think there is a capability issue that we will have to address in terms of continue to be able to treat everyone. So thank you.
Thank you very much, Emma. We have time for one quick question. Here in front.
Very nice, Emma. You mentioned antenatal screening. In SMA and DMD, there are things that we want to get treated sooner. Shouldn't we be doing antenatal screening? The technology can be sharpened, and it's not difficult to give treatments antenatally. It's being done for multiple diseases now. Seems like these diseases are ideal ones to push for. So newborn screening is not soon enough.
It's not soon enough, certainly not for the 2 copies. So as you know, OBGYN now, they're offering screening, prenatal screening for a number of big panels, and SMA is that. So there are many, many families that come to us because they know even before newborn screening that they're carrying a baby with SMA. That's the opportunity that we can leverage to really have induced delivery at 35, 36 weeks as soon as possible and really treat very, very quickly. And I think there is more that we can perfection about that. And I think the early delivery, you know, in the early days, we couldn't treat before 40 weeks. There were policies that we were able to actually change. So absolutely, prenatal screening. And then the idea of being able to treat in utero, which for gene transfer remains quite impossible at this point, but there are cases, at least one case that I know of, where mom carrying a 2 copies baby was actually allowed through an IND to take risdiplam for 3 months prior to delivery. And I think we're gonna see more of those type of strategy.
Thank you again, Emma. I'm happy to introduce our next speaker who's coming up to the front. This is Scott Harper from Nationwide's Children's Hospital, and he's going to provide us an overview of potential in genetic therapies in FSHD.
Good morning. Thank you for inviting me. I'm happy to be here. And also, that was a great first talk. It sort of set the stage for what I'm going to be talking about. That's the very last slide. Okay. So if I can just make a comment too, I think the point about the previous speaker made about looking at empty versus full capsids with the AAV vectors that we're using to deliver gene therapies is important. And I would also add to that I think we also need to be looking at the consistency of genome integrity. I'm happy to talk about that later, but I think what we're finding, if we sequence these AAV products, they're not uniform. We're getting a lot of mixed genomes. So I'm going to tell you about FSHD today and some of the work my lab's doing within my academic lab and also with my company that I started a couple years ago. It's called Armidas Bio. That's the major disclosure I want to highlight here. I'll be showing you some data. Some of you in this audience have seen this, but we have some new data that's helped to de-risk the programs that we're developing. And I'll just begin with my entree into the FSHD field, which was in 2007 as a new assistant professor, and this was the state of the field. We didn't really know much about the disease. The pathogenic mechanisms, we were sort of touching on them, but we didn't have a disease gene that the field was focused on. We had limited patient cell lines, and actually some of those early cell lines turned out not to be FSHD cell lines. Because we didn't have a mechanism that was defined. There were no animal models that were at least published, and as you might imagine, no treatments then. So as a new assistant professor, this is maybe kind of a dumb idea to start a lab when we didn't know anything about this disease. But it's been exciting to work in the field, and I would argue that FSHD is the most, among the most, if not the most complicated genetic diseases. And unfortunately, because this is only a 20-minute talk, I don't have time to give the whole mechanism. I think that probably the field knows, the folks in this room understand the disease. So I'm just going to have to give you the punchline about the mechanism. And that is that there's 2 major things I want to highlight here. FSHD requires hypomethylation of the DNA repeats called D4Z4 repeats located on chromosome 4q. And that hypomethylation, which can occur by a couple of different mechanisms, leads to derepression of a gene called DUX4, double homeodomain 4, or double homeobox 4. So DUX4 is the primary FSHD gene, and we and my lab in the field are focused on DUX4 inhibition as potential therapies for FSHD. And so what DUX4 produces is a transcription factor that's normally expressed in early development. And it's not supposed to be on in skeletal muscle. When it becomes derepressed and turned on in skeletal muscle, you develop FSHD. So one of the problems with FSHD and DUX4 in particular is that it's very rare. And so you would think that, I'm sure many folks in this lab have FSHD patients, and some have very significant disease. And so you'd think that the disease gene that was associated with that, that's causing that muscle damage, would be easy to find. And it actually turns out it's not. It's pretty rare. The current estimates are 1 in 1,000 to 1 in 10,000 myonuclei at any given time express DUX4. So just doing the math, that means that some myofibers in a big muscle might not even have a single DUX4 positive myonucleus at any given time. Seems like there's some timing on my slides that's moving them forward, and I'm not trying to. So anyway, what does this mean? This has implications for therapies, how we think about doing therapies, but not just developing therapies, but also doing clinical trials. It becomes a difficult thing if you're trying to do a clinical trial and you want to demonstrate target engagement and you can't detect the target in a human being. So I want to give you a quick overview of what the field is doing. Now, this is a busy slide, and then I'll tell you about some of the work that we're doing in my lab and in our company. This is a busy slide, but I just want to highlight a few things, which is that what this shows is essentially the gene expression pathway, the central dogma of molecular biology, right? That our DNA encodes genes that can be transcribed into messenger RNAs, and then they become processed to serve as templates to make protein. And so there's a number of different steps in this pathway that I've noted here. And the important thing for FSHD is that because we're trying to eliminate DUX4, or at least inhibit its expression, this is the pathway that every muscle cell requires to produce DUX4, and it gives us opportunities to intervene at each one of these steps. The work I'm going to be telling you about today from my own lab is going to be focused on at this level, this post-transcription level, and I'll get into that a little bit more. But the field is actually, both in academia and in industry, are touching upon all these different steps in this pathway. And I'm going to tell you about gene therapy. The previous speaker did a beautiful job setting this up, so made my job a little easier. But I teach a class at Ohio State on gene therapy to PhD students, and I think we all think we know what gene therapy is, but when you ask students to define it, they can't come up with a good definition. So this is the simplest definition that I can come up with. Treating disease with nucleic acids. So this means either adding back genes that are missing, or in our case with FSHD, reducing expression of those genes. And I'll tell you about how that's done. But the way we're doing this is with using adeno-associated viral vectors as a delivery vehicle to deliver our inhibitory gene cassettes that can knock down DUX4. And the AAV vectors were already mentioned. I'm going to tell you a little bit about the progression of our program using several different AAV serotypes. But the idea here is that we can package a therapeutic inside an AAV vector. We can grow that in a lab or in a manufacturing facility, deliver that to a human being. In our case, we're thinking systemic delivery to hit muscle. And then that vector can find muscle, uncode itself, and deliver the viral DNA to the nucleus of the target cells. And the beautiful thing about AAV is that in post-mitotic cells like muscle, the genome doesn't integrate, but it can remain in the nucleus, essentially, at least theoretically, forever. Now, that muscle, if it turns over, you're going to lose that AAV genome, but at least theoretically, one delivery should produce long-term durability. Now, this is sort of an overview of the field, and I'm not claiming that this is 100% comprehensive. But this gives you a snapshot of some of the things that are happening in the field, targeting DUX4 at different levels in that pathway that I showed you. I'm going to talk about mostly this RNAi therapy that we're doing with Armidas Bio here. And if I have a little bit of time, I have a couple slides on a CRISPR project we're doing as well at the end. So let me tell you about what we're doing with what we're calling RNAi-based gene therapy for FSHD. Now, this is a project that initiated in my lab many years ago, led by Lindsay Wallace, who's shown here. She continues to lead this program. This is now part of Armidas Bio. And what I'm going to give you a few keywords here, but a couple of the main ones are MI405. This is what we call our therapeutic. It's a microRNA that we've engineered to knock down DUX4. I'll tell you a little bit more about that in the next couple slides. And then we're cloning the genome, that expression genome that produces the microRNA inside an AAV vector and then delivering that to, in our case, mice and hopefully humans soon. And then we've developed an animal model called the tick DUX4 model. I'll show you a little bit about that. And we've also done some work in a xenograph model. So this is a mouse model that contains FSHD patient myoblast. We did this in collaboration with the Emerson lab at UMass. I don't have time to show that data, but I just wanted to mention that we've used that model as well. So what are we talking about with RNAi therapy? So, I think gene therapy in general, and actually all the approved systems so far have been gene replacement. So a gene is missing, you have a recessive disease, and you want to add that gene back. And that's what happens in SMA, that's what's happening in DMD. But as I mentioned, DUX4, or FSHD, is caused by de-repression of this gene. So adding back more DUX4 is actually going to make the disease worse. We want to eliminate DUX4. So how do we do that? Well, we're using RNA interference, also abbreviated as RNAi. And this was discovered in worms in 1998. There's a Nature paper in 1998, and only 8 years later, the discoverers of that mechanism won the Nobel Prize. So this is a natural mechanism of gene silencing. All of us sitting in this room have over 1,800 natural microRNA genes in our genomes. And so our cells produce these, and they function to regulate the expression of other genes. So what that means is gene knockdown or gene silencing. Those are words you might hear associated with RNAi. And in particular, these microRNAs, they don't code for protein. So they're not protein coding genes. What they do is they form these hairpin structures, and I've got one shown here. The hairpin structure, and I don't have time to go into the biology of it, but the portions of that sequence located between the arrowheads is the functional part of the molecule. And so every natural microRNA has a certain sequence that it uses to base pair with other messenger RNAs in a cell and regulate those, silence those. We can take that biology of natural microRNAs, we could switch out the sequence in the middle, so this part that's underlined, and retarget these microRNAs to any gene we want to silence. And in this case, we're doing that with DUX4. So we've engineered these artificial microRNAs to retarget them to DUX4 and cause it to be silenced. And so this happens at the RNA level here. And here's the general idea, and then I'll show you some data. We take a natural microRNA, in this case miR30, we re-engineer essentially the guts of the molecule so we can retarget it to the gene of interest, in our case DUX4. We clone that into an AAV vector. We grow these vectors in the lab, deliver these to mice, in our case, and again, like I said, hopefully to humans. And then we have this whole process where the AAV vector becomes unpackaged. We have the genome produced in the nucleus of the target cell, and when it's there, it can constantly produce our therapeutic. So one-time delivery, the genome is there. The microRNA that regulates DUX4 is now being cranked out by the nuclei of those muscle cells. And like I said a few minutes earlier, theoretically, so long as that muscle is not turning over, that should last indefinitely. Now, we don't know, we don't have the real-world data on that yet. I think the longest anybody's looked is about 10 years. So I'm going to show you a snapshot of some data. Some of this is published, some's not. This is this is where we developed our first microRNA, or our lead microRNA. We call it MI405, as I mentioned. And we cloned a number of different sequences. We were trying to develop the best therapeutic we could find. So we ended up screening 42 different microRNAs that we designed. MI405 became the lead because it could suppress DUX4 extremely well. So what we show here is a Western blot. This is just protein showing here that we can eliminate DUX4 protein. This is in vitro compared to controls and some other sequences that don't work as well. This is endogenous DUX4 in a human FSHD myoblast, or myotube culture, actually. We've stained this with RNAscope, and what that does is it can light up messenger RNAs in a cell or even in vivo, and we use this brown stain to detect it. So every little brown dot here represents one DUX4 messenger RNA in this set of myotubes. When we treated these myotubes with our therapeutic, we didn't find those at all. We were able to knock that down, and this is in vitro. And this is also just demonstrating that we've done a number of other things, that that microRNA is able to suppress the toxic effects of DUX4 in cells. So DUX4 causes apoptosis, in human cells in vitro, and we can prevent that from happening. We can measure that by a caspase assay. And we can also look at DNA damage. DUX4 causes apoptosis leading to DNA damage. There's something called a COMET assay, which is pretty fun to do in the lab, where a cell that contains nuclei that are damaged shows its DNA, it looks like a comet here. And so if a cell is dying, it's got this comet shape. And you can quantify it's called the tail moment. And so we also show here that we can suppress that with our microRNA. So in vivo, we at the time we started developing these things, there weren't any mouse models, as I mentioned. We had to make one. Some other ones have come along since, so there are several models in the field now. I'm going to tell you about the one we made. We call it TIC-DUX4. TIC stands for tamoxifen-inducible Cre recombinase. And so I don't have time to go into the specifics of this, but I'll just say that DUX4 is toxic in developing mice, and you can't just turn on DUX4 through embryogenesis and expect to have viable embryos. You have to regulate its expression. You have to turn on DUX4 after the mice develop. So we use this system to allow us to turn on DUX4 when and where we want using tamoxifen. So this is a tamoxifen-inducible system. You literally give the mice tamoxifen. Here's a PhD student of mine giving tamoxifen to this mouse by oral gavage. And when you give tamoxifen, it turns on DUX4 only in skeletal muscle. So this allows us to create viable mice and then turn on DUX4 at a later time after they've developed. So here's a little bit of data in vivo. So there are two different pieces of data here, two different experiments. So the other thing I want to say about this is I mentioned that in an earlier slide, I mentioned that DUX4 is really rare and in humans. And so for us to be able to produce an animal model that actually has some phenotypes, we're expressing DUX4 more abundantly and at higher levels in the mouse than is seen in humans. So we call that our moderate DUX4 expression level. And what that does is it produces abundant, widespread muscle damage. But there's also some leakiness to the system. So if you don't treat the animals with tamoxifen, there's just a little bit of leakiness that sort of mimics what is seen in patients. So you have this rare burst of DUX4 expression that produces damage over time. And so we've taken advantage of this uninduced system, which is the slow leaky system, which we think is more physiologically relevant, and then also the moderate system, which, where we can drive expression and drive pathology, which I think is to be able to contend with that phenotype, I think bodes well for what we're trying to do. And so here's the moderate, where we've got higher levels of DUX4. You can see the kind of damage that occurs in the muscle at 8 weeks in this mouse when we've induced DUX4 tamoxifen. But here's the mouse that's been treated. We can get complete protection. And we've taken this out to a year in the leaky model. And what this shows, the blue, this is a low power shot of a mouse muscle, and we've intentionally colored the damaged myofibers blue, just so you can sort of see from a big picture standpoint the kind of damage that's produced after a year of expressing DUX4 at low levels. And we have, after 1 year, after a single injection in a mouse, we have complete protection after a year. Now, this is with the first generation adeno-associated virus, okay? So this is AV6, and then we later pivoted to AV9 because AV9 had been used in the clinic with Zolgensma, and we wanted something that had some clinical relevance and some safety data. But as was mentioned in the previous talk, those first generation vectors, to hit enough skeletal muscle, require very high doses. Is it possible to turn on this video? Oh, thank you. Okay, so one of the outcome measures we did as a functional outcome measure, we were trying to do a dose finding study with our AAV vectors to ask how much of our vector do we actually need to deliver to a mouse and then later to a human to have some benefit. And so we used this ambulation assay as an outcome measure. And it's a very simple assay. It's pretty cool. We have these cages that have lasers in 3 dimensions, X, Y, and Z direction. And as the mouse is running through their cage, it'll trip a laser. So a mouse that is normal or healthy can move around, and it trips these lasers, and the computer counts it. Mice that are sick don't move around as much, or they can't rear back on their hind limbs as much, so they don't trigger as many lasers. And so the data then becomes pretty well quantifiable. And so when we did this work with our first generation vector, AV9, expressing our MI405, this is messy data, because these mice are quite variable actually. We saw that to produce a phenotype, we had to induce over 10 weeks high levels of DUX4 with tamoxifen, and the black bar shows saline-treated animals. They decline and sort of stay down. And then we tried 3 different doses, 6E13 vector genomes per kilogram up to 3E14. Now this is the dose range that had been tried in clinical trials for other neuromuscular diseases with this system. And at 3E14, we could rescue this phenotype. And again, I want to remind you that we were really driving the pathology here. We don't see any kind of ambulation or functional deficits if we just use this leaky expression system. We have to turn on DUX4 at high levels. So this was, at this point, we were excited about this, but then some of this news started to come out that I think everybody in the room is familiar with, which is that these high doses were associated with patient deaths. And we started a company, we're trying to get funded, and the reviews we're getting back from venture capital firms or pharma companies were, well, we don't want to fund a program where that's this risky, where you have to have such high dose. So we started looking for, despite the fact that in our mouse models, we didn't see any evidence of toxicity of the high doses. So we started looking for a second generation AV capsid. And solid was mentioned earlier, SLB101. We tested a number of different capsids. And the goal was essentially these things. We were looking for something that could give us about a 10-fold, roughly, in the 10 to the 13th vector genomes per kilogram range, which had been so far been proven pretty safe in the clinic. This is sort of what the field has been going to. Muscle targeting, liver targeting. So this was intended to be safer and cheaper. And then we also wanted to have something that had been de-risked translationally and clinically. And luckily, we were able to form a partnership with Solid. We licensed their capsid that was already mentioned, SLB101. This is actually in the clinic now. They're doing a DMD trial with this right now. And so we were happy to be able to get access to that capsid. And now we've been wrapping up a study now with SLB101 in our microRNA. And this is just a couple of functional tests. Here's this is the open field test, which is the one I just showed you again. And we're able to see rescue. We've only taken this out to 6 weeks now, not 10. But we're able to see rescue in a couple of different open field studies. Here's the total distance. So the green represents wild-type mice. The red represents saline-treated TIC-DUX4 mice. So these are FSHD mice. And then the blue represents those treated with our solid vector at 6 times 10 to the 13th. So now we're in that safer range. And we're able to recover, okay, almost done. We're able to recover function there at lower doses, which is what we were hoping for. We're also seeing increased distance on a rotarod. And the great thing is that this demonstrates biomarker activity. So we've also looked at molecular outcomes. There's a biomarker in mice called WFDC3 that is a DUX4 activated biomarker. So if we can knock down DUX4, we see this reduction in WFDC3. And so I mentioned 6E13 is where we were seeing our functional benefit with the solid vector, but even at 3E13, we're seeing suppression of DUX4 and that biomarker and protection histologically. So we think our MED going into the clinic is going to be 3E13 to 6E13 vector genomes per kilogram. I'll just mention that we've dosed almost 1,000 mice, and we're seeing consistently great data with this. Now we're happy that we have the solid vector. And I know I'm out of time, but can I give one? Okay. Okay, so I'll do this briefly. I have one more story I want to tell you. It's just a quick story. It's related to CRISPR and, at least in our hands, the viability of using CRISPR in vivo. So I would say that, here's my summary. During the last 15 years, FSHD has become a viable target for therapy. I think the field surrounded DUX4 as the target. We want to inhibit DUX4. There are a number of different approaches in the field, including the ones I just told you about. Our MI405 triggers potent RNA interference against DUX4, and now we have a capsid that can deliver it at lower doses that we think are in a safer range. And we think that we can use this same idea to go after other dominant neuromuscular diseases. And here's my bullet point on the CRISPR. So this is work that was done by Afrouz Rashnadajad, who used to be a postdoc in my lab. She now has her own lab. And what we're doing here, we're using a CRISPR enzyme called CRISPR-Cas13, which cuts RNA. It doesn't do genome editing at the DNA level, it cuts RNA. And so Afrouz developed a CRISPR-Cas13 system that was functional to knock down DUX4. And the reason we pivoted to this, just as an experiment, was because it was suggested that CRISPR is more beneficial or more potent than RNA interference. So we tried it, and it works. But what we're finding actually, and it works actually within a few weeks, we see improvements. But what we found that over time, we're seeing immune responses to the Cas13 protein, in particular CD8 immune responses, cytotoxic T cell, only not to the AAV, but only to the Cas13, and then we're seeing loss of efficacy over time, and this is happening in a mouse. This is not unprecedented. Dongsheng Duan had published this in DMD dogs with Cas9 a few years ago. So we think that this is important. We're writing this paper up and hopefully going to be publishing soon. Thanks for giving me an extra couple minutes to show that. That's it. Thanks.
Thank you, Scott. It's probably a good thing that there's so much progress that you can't tell it all in 20 minutes.
Yeah, thank you. Are we no questions or?
We have time for one question. Maybe do the question in the back over there. Oh, sorry, the microphone is up in front. Go ahead.
So I don't think the DUX4 hypothesis is settled myself.
I'm sorry.
I don't think the DUX4 hypothesis is. I think it's necessary, but not sufficient.
Okay.
There's a lot of evidence that it may not be the whole story. So we had one of my most neglected papers. I have lots of neglected papers. Showed that the phenotype in FSHD is inherited over and above just the presence of the gene that runs in families, and the phenotype the variability of the phenotype just can't be explained, I don't think, by the stochastic effects of a single mutation. There's another genetic factor, I would submit.
I agree with that. I think the field agrees with that too. There are definitely modifiers, and I mentioned at the beginning that there are a number of triggers of the epigenetic changes, and we know that there are DUX4 interacting proteins now, so there are probably a lot of different factors that are influencing the penetrance of that. But I think the field, so I would respectfully disagree that DUX4 is not the primary gene. I think that there are probably other things contributing to that, but DUX4 is absolutely involved. I guess that's what you said, necessary but not sufficient. So we're agreeing. Yeah. Okay, thank you.
Okay, I'd like to welcome Mario Saporta from the University of Miami, and Mario will be speaking today on genetic therapeutics in neuropathies and CMT.
Sorry, I have to interrupt. We have to get Mario's slides again, so can we go to the next talk and then come back to Mario? I'm very sorry, Mario.
Okay.
My mistake. I thought we had it, but we couldn't find it. Okay. So could we introduce Mr. Thornton next?
Is Charles Thornton? Oh, yes, great. Okay, so we'll stick to muscle then. So thank you, Charles Thornton from University of Rochester, and Charles will be speaking on genetic therapeutics in myotonic dystrophy. Thanks, Charles.
Good to be here. Thanks for the invitation. So even before we knew anything about the mechanism in myotonic dystrophy, there was one hopeful note. It's not a field that's saturated with hope, I think you'll agree. But just by looking at the histology, the main feature is that there's a lot of trouble with the muscle architecture. There's shrinkage. There's not a lot of fibrosis. And in muscles even that are moderately weak, the pathology is not so severe, really. The main feature late in the disease is fiber shrinkage. Without a lot of fibrosis to limit response, the idea was that if you could eliminate the trigger of the disease process, that you might get a very impressive recovery. And then the gene was discovered, and it was an unusual mutation, and it had two troublesome features, and one of them was that this repeat in DMPK keeps getting larger the longer that a person survives. And people start with hundreds typically, but by the time they have symptoms, they usually have more than 3,000 repeats in the DMPK gene in skeletal muscle and probably in heart and brain as well. And the other troublesome feature was that this gene is made into RNA, and that creates an RNA sequence with a long repeat, a CUG repeat, and that RNA forms clumps and it interacts with proteins. And so when this was seen, that these clumps form, this was our first glimpse of the enemy, and the clumps are shown here in red in a nucleus stained blue in a muscle fiber stained green. And the reason that they cause trouble is that there's a group of RNA binding proteins called muscle-blind proteins, or MBNL proteins, that unfortunately have a very high affinity for this CUG repeat sequence, and they interact with it extensively. And the effect of that on the binding protein is that its location in the cell is completely shifted. And these are high-power views of nuclei from muscle or heart, and normally the muscle-blind proteins, which have an important function in the nucleus, which I'll get to in a minute, are distributed throughout the nucleus, but in the disease, they're completely drawn into these clumps, both in heart and muscle and also in brain. And the function of muscle-blind that we know the most about is that they control alternative splicing. And let me just remind you what alternative splicing is about. So here's a hypothetical gene. It has 3 exons, but there's some flexibility about how the second exon is handled. And when the gene is converted into RNA, in some of the primary transcripts, this exon is skipped over by the splicing machinery, but in other transcripts, it could be included. And muscle-blind controls that decision. So this is a gene that can produce 2 flavors of RNA and encode 2 different proteins. And sometimes the functions of these proteins can be very different. And here's a favorite example recently, which is the effect on the splicing of the voltage-gated calcium channel in skeletal muscle. So this is a protein familiar to all of you who work in muscle. It's essential for muscle contraction. Point mutations in this gene are the cause of periodic paralysis or congenital myopathy. But the problem in this case is with the splicing of this exon 29. And in healthy people, this exon is almost always spliced in. About 90% or 95% of the transcripts contain the exon. But in people who have myotonic dystrophy, this exon is variably skipped. There's quite a good correlation between how weak the muscle is and how much skipping of this exon there is. That exon encodes a small domain on the extracellular surface of the channel, and when that domain is missing, then there's increased calcium entry with every action potential. And that turns out to be somewhat deleterious on its own right, but it's particularly harmful when it's combined with myotonia, which causes too many action potentials. And the myotonia relates to a problem with the splicing of a chloride channel. I'm talking to you now about splicing of a calcium channel. And if you make both of those changes and engineer them throughout all the skeletal muscles in a mouse, and these are the only splicing effects in the mouse, then that's lethal by 15 weeks of age. And it's also rescuable to a pretty large extent by calcium channel blockers. So from a therapeutic perspective for clinicians, this is a dream therapeutic scenario, I would argue, because the muscle-blind proteins are there in the cells, they're just in the wrong location, and if they were released, they can and do resume their work, and then you could harness the tremendous regenerative potential of skeletal muscle, and perhaps the regeneration wouldn't be limited too much by fibrosis because there doesn't seem to be a lot of fibrosis. And the options for doing this in the clinic now that I'll talk about today are mostly genetic therapies. Now, these are genetic therapies that aren't gene therapies. The top three of these are all synthetic small bits of DNA or RNA-like materials. I won't talk about gene therapy vectors today. The strategies mainly now focus on antisense oligonucleotides. I might call these ASOs, or short interfering RNAs, siRNAs, or a different chemistry of the modified DNA, which is called a morpholino, or sometimes called a PMO. And the first of these are meant to remove the toxic RNA. And here there's another fortunate feature of myotonic dystrophy, which is that it seems to be, at least in animals, in mice, you can completely remove DMPK protein, and there's no phenotype that's been observed yet. Knocking down DMPK in non-human primates also seems to be very well tolerated. So this is not a situation where if you're trying to get rid of a toxic gene, silence it, that you have to be terribly concerned about the effects on the wild-type allele in this dominant disease. The objective with the morpholino, the blocker oligo, is to just block the RNA so that it can't interact with the binding proteins. And let me go through the second of these first with the blocker oligo, because this is the one actually that we worked on first. And we worked on this actually not because we thought that this would ever turn into a drug, but just as a proof of concept that if you could block the muscleblind and release it into the nucleus, that it would rescue the phenotype. We thought that this was just a artifice, though, blocking it this way, because there was no hope ever of getting this type of material into muscle cells, but it was worth trying just as a proof of concept for the approach. And so now what we're doing is we're making a morpholino, a blocker oligo, which will bind to the repeat RNA just according to normal hybridization principles. It's a CAG repeat to bind to CUG repeats, and hopefully just tile across this repeat tract in the RNA so that the binding proteins don't get stuck on it any longer. And if you did this, then the muscle-blind proteins would hopefully be released into the nucleus and resume their function. And by injecting this morpholino directly into muscles, one muscle, this is a one muscle treatment strategy here, of a transgenic animal, it has a large amount of this repeat toxic RNA shown here in red. You can completely disperse the foci, and it restores the distribution of muscleblind, and it corrects the splicing changes, and it eliminates the myotonia. But this was injection of a single muscle. If you inject this intravenously, nothing happens. Not enough of the PMO gets into the fibers to have an effect. And then moving back to the antisense oligonucleotide strategy, the ASO now is designed to bind to the RNA, not in the repeats, but upstream or downstream in the other parts of the DMPK transcript. And once it binds, there's an enzyme that exists in all cells. It's called ribonuclease H that sees this unusual hybridization, this complex of a DNA-like substance with RNA. And where it sees that combination, it cleaves the RNA and makes a single cut. And after making that cut, these can be recycled, but this RNA becomes unstable. It's rapidly degraded by other machinery in the cells, and the muscleblind is released. The strategy with the siRNA is exactly the same as you've just heard about from Scott. And it's surprising how quickly these things can work in mice. Mind you, this is with an aggressive dosing strategy beyond what could be used in a person. But with a high-dose treatment regimen in a mouse, giving the ASO twice weekly by subcutaneous injection, you can start to see a reduction after the first injection, and you get a strong reduction after the first week. And the correction of the splicing, which is shown here by measuring splicing changes across 35 or so different RNAs and averaging them together to get an overall splicing index, which should hover around 0. and it's abnormal in the mice to begin with, but the correction of splicing pretty much exactly parallels the reduction of the toxic RNA and the rescue of the myotonia, which is now shown here on a very crude qualitative scale on the vertical axis, where 3 is a lot and 0 is none. The myotonia goes away almost as fast as the splicing is corrected. Now, the prediction of this result from a trial is that there's the potential that if the material accumulates in muscle to a concentration that's actually active, that you can get a very rapid correction of the splicing and the myotonia. But I think in this scenario, the myotonia is an outlier, and there's some technical reasons for that we could talk about. We can't really expect that to happen with the other muscle features necessarily. They may take much longer to correct, but we don't know very much about that yet because we don't have good animal models that actually recapitulate the other features of myotonic dystrophy in skeletal muscle very well. But the data were encouraging enough to support a clinical trial, and the question was, by injecting the antisense drug, this was subcutaneous injections again, systemically, could you get enough of the drug into muscle tissue to have a strong effect? And this is a trial carried out by Ionis, and they had a goal in mind, which is the amount of oligo that was required to get a strong effect in non-human primates against the DMPK gene. And they fell short of getting a concentration anywhere near that high. They were several fold below the goal. It's not that the material did not enter muscle. It did to some extent, and it could be measured there. There were a few people who seemed to have a response, but it was an impractically high dose. And so this was a very well conducted clinical trial, and it helped really inform the field that the problem with this was that we had to get delivery into the tissue better. And the solution to this really follows from the TTR amyloidosis example, which is conjugate the oligo to a carrier molecule that can then deliver it into the right tissues. And the surprising thing is that once biotechnology companies saw what the problem was and focused their energies on overcoming it, that there's a remarkable diversity of solutions about how to get oligos into skeletal muscle that I think will be very applicable across many diseases in the future. And these are the main strategies that are being used now. One is monoclonal antibodies or fragments of monoclonal antibodies. Another is peptides, arginine-rich peptides or cyclic peptides, and lipids are also being used. And all of this work was going on simultaneously in several different companies, and now 5 of these agents are currently in clinical trials, and the one that's furthest along is the siRNA strategy developed by Avidity, and a pivotal trial has just started for that. And it wouldn't be proper for me to say much about the results from these early phase trials. That should fall on the companies and the principal investigators who are mainly in control of those trials. My role in that is just supportive. I enjoy seeing the patients. But I will just show you just a glimpse of some data from public presentations. This is not peer-reviewed by Avidity, just to give you a flavor of why people are excited about this, and that this shows that, and keep in mind that these are a very small number of patients treated here in a phase 1/2 trial, 9 people per group. These doses here examined. There is reduction of the target DMPK mRNA, and there's also an improvement of alternative splicing, but the effects are not simply biochemical effects. And as expected from the mouse models, there is a surprisingly rapid improvement of myotonia, and it's it's unlikely that this will just be an effect on myotonia. And these are 1-year data. So this is not against placebo controls, but against natural history controls. So that's always a caveat about how the controls were selected. But in any case, these data do suggest that there's an effect after 1 year on muscle strength and muscle function and also quality of life. The very encouraging thing is that we have a great set of solutions for the delivery problem, and we have a good set of payloads to deliver into muscle. So I think the prospects for having a group of approved treatments for myotonic dystrophy in the next few years are looking strong. It's far too early to know which of these drugs is most effective. There's a lively debate about whether ASOs or siRNAs are the best way to get rid of the toxic RNA. In general, siRNAs tend to be more potent, but there's a question about whether they work as well in the nucleus, which is where this RNA spends its entire life. The blocker oligos work very well in a transgenic mouse that has relatively short repeats, 220 repeats. There's a question of whether they will work as well when the repeats are 10 or 20-fold larger than that. But I think looking forward for the NMSG community, we should plan for the likelihood that there's going to be several approvals and that there's going to be a need for a whole lot of post-approval research by investigators of the NMSG. And I think it's likely that we'll come through these trials and through the approval process still with big questions about which treatments are right for a particular person, what their effects are in the non-skeletal muscle organs, when they should be started. Should they be started before the symptoms begin will be an important question. And what happens to the repeat size while the RNA is being dealt with? Will the repeats continue to grow? And if so, what are the risks that might be associated with stopping treatment? Would that actually return a person to a disease state that was worse than the one that they started? Many interesting questions. So I just want to close with these acknowledgments and particularly acknowledge my colleagues in the Myotonic Dystrophy Clinical Research Network and people at the University of Rochester who worked on this. Thanks very much.
Fantastic. Thank you. We've got time for maybe 2 questions. If I just start, I mean, based on your experience, I know it's difficult, but if you had to put your money on, is it a particular pathway or option, or do you think it's going to be a sort of cocktail of genetic therapies together? What do you feel?
Oh, you know, it would be completely guesswork. So I know that that's an important question and people are asking it, but could I respectfully decline? Because I have no idea which of these is going to work the best. And you know, in fact, let me just go one step beyond that and say, I think that they're all going to work pretty well, and that we will be in the universe of trying to, you know, lay down a certain level of therapeutic response and then see how much better can we do. These drugs are having their intended clinical and biochemical effects. I don't think we've seen yet that they correct all of the splicing changes, and we know that that can be done in a mouse model, and so there will be incentive, I think, to keep pushing the envelope on this and try to get things that work better.
Thanks, Charles. Very nice talk. Just a quick question. You sort of prompted it about the siRNA approach in targeting a nuclear RNA. Can you expand on that a little bit?
Well, I can kind of frame for you the debate that's taking place, and there's strong opinions on both sides. And there's one school of thought that the machinery for siRNA really isn't found in the nucleus to an appreciable extent. And I think that that might be true if you're trying to detect it by immunocytochemistry or something like that. But there's actually many lines of evidence, though, showing activity of siRNAs in the nucleus. And from my perspective, even the early phase trial data from the Avidity trial convinced me that there's definitely activity in the nucleus.
One
So
question.
how do you fit the maternal versus paternal myotonic dystrophy phenotype in your hypotheses about the causation?
Well, I think there's a lot of questions about this really, it's rare to find congenital myotonic dystrophy with paternal inheritance. So I think that one answer to that certainly is there's a difference in the expandability of the repeat when it's passaged through the female germline compared to the male germline. It's not rare to find a family with a mom who's got 80 repeats and a child who's got 1,200 repeats, a huge increase in one generation, and you virtually never see that in men. So I think that's probably the main explanation. Thanks.
Right. Thank you very much. Thank you, Vino, and the planning committee for this invitation. So yeah, we're going to switch to peripheral neuropathies, and I have a very kind of hard task because CMT is not a single disease, so I'm going to basically cover everything that the previous three speakers introduced and discussed, so that make my job a little bit easier, but not that much. Those are my disclosures. And I just wanted to put genetic therapies for CMT in the context of a larger pipeline that's now ongoing. Those are therapies that are now under development by industry for CMT and different types of CMT. And in the blue here, and I'm going to make it a little larger, those are all genetic therapies for CMT1A. So there's a very kind of speed up of this process of development and interest by industry for therapy development for genetic therapies for CMT. So as I mentioned, CMT is not a single disease, and we're going to try to cover at least most of the different subtypes. But when I think about genetic therapies for CMT, I think about targets, strategies, and challenges. For targets, we're going to be covering three main types. One, a gene dosage issue of PMP22 overexpression in CMT1A. Then loss of function mutations in recessive CMT and other examples like X-linked CMT. And then gain of function mutations where there's a dominant variant causing a toxic gain of function that we'll have to deal with when developing therapies. Strategies, I think most of them have been covered. So ASO, siRNAs, microRNAs, gene replacement therapy, and then I'm going to talk a little bit about allele-specific knockdown for the gain-of-function mutations. I'm not going to talk too much about knockdown and replace just because of time, but we can discuss this further. And then we're going to discuss some of the challenges, which are common to other diseases, and some of them have been discussed already. Right, so starting just a brief review of CMT. So CMT is the most common neuromuscular genetic disorder, affects about 3 million people worldwide. It is a length-dependent neuropathy in most instances, causing distal sensory loss and weakness with the typical foot and leg atrophy and change and morphological changes. It is either primarily or as a secondary process, a process of axonal degeneration. And axonal degeneration seems to really drive the functional impairment in those patients. And as I mentioned, it is a very genetically heterogeneous disease with now over 100 different genes being linked to this phenotype. So one can make the case that CMT is actually probably over 100 different diseases, and they will all have a different genetic therapy approach. But they can all be clumped together with either a primarily myelin-related gene or primarily axonal gene, so that's how we classify CMT initially as type 1 and type 2. And at the end of the day, it seems like either the myelin or the primary axonal problem will affect how axons are functioning, transporting cargo, and connecting to the neuromuscular junction. But I would like to make the case that CMT is probably, from a genetic perspective and a biological perspective, probably over 1,000 different conditions if you think about the different point mutations causing different biological responses. And that's very true for some of the most common types of CMTs, like CMT1B, 2A, 2E, where we see from animal models and basic research that depending on where in the gene the mutation is, the biological process that's triggered can be very different. And in any case, from forgetting about gene replacement therapy, but just trying to target some of those genes where multiple missense mutations happen, the design of some of our strategies will have to be very customized to specific patients. So now we're going to switch to CMT1A. So CMT1A is a disease linked to an extra copy of PMP22, so it's a kind of overexpression disease, and most of the therapies that are being developed right now are oligonucleotide-mediated PMP22 downregulation. So just quickly reviewing CMT1A. CMT1A is caused by this 1.4 mega-base duplication on chromosome 17 containing the PMP22 gene. It is really what we think about CMT from a phenotypic perspective. So patients develop normal early on, they achieve normal motor milestones, and then by childhood, at some point, either early, mid-childhood, they start to develop issues with balance, ankle stability. They are considered clumsy kids, slow runners, bad at sports, and then eventually they will present the distal muscle atrophy and sensory loss, and also the foot deformities. It accounts for about 70% of all demyelinating cases and about half of the CMT cases that receive a molecular diagnosis, so it's a very common disease from that perspective. And based on several rodent models overexpressing PMP22, we are now pretty sure that it is the overexpression of PMP22 that's the cause of the neuropathy and the demyelination. So those are kind of the main avenues for therapy development for CMT1A currently ongoing. The initial work has been focused on the use of ASOs delivered subcutaneously, targeting the PMP22 open reading frame and tested in the CMT1A rat and then the CMT22, the C22 mouse. Then a couple of works using siRNA conjugated with different molecules to allow for peripheral nervous system delivery, both delivered intravenously, and then a more recent work using a microRNA approach, and that works from Scott Harper and Cleopas Cleopa from the Cyprus Neurological Institute, using an AV9 to deliver the microRNA, and they tested that on the C61 mouse. So as you can see, there are different models of CMT1A, and that makes things a little bit confusing and harder to compare. And that has to do with the number of PMP22 copies that each mouse carry. Ranging from 3 copies on the mouse, on the C3 mouse, then 8 copies on the C22 mouse, and 4 copies on the C61 mouse. So they are a little bit different from a biological perspective, and they don't completely recapitulate the human genetic of a single PMP22 duplication. So I'm just going to very briefly summarize the main results from those, at least those 3 different approaches. So for the ASO-mediated PMP22 downregulation, they achieved about 40 to 50% PMP22 mRNA reduction when they administered the ASO. That translated to a partial behavioral rescue seen here with grip strength and time in the rotating rod. So they did improve, but they did not reach wild-type levels. The motor nerve conduction velocity was rescued to about 80% of the wild type from what usually is less than 50%. And then they see an increase in myelinated fiber density. So they're very promising results for the ASO. Looking at the siRNA-mediated PMP22 downregulation, this is work from DTX Pharma. They use a conjugation with a lipid molecule to allow for better delivery to peripheral nerves. They achieve about a 50 to 70% PMP22 reduction. Again, a rescue to about 80% of the motor nerve conduction velocity. And what's very striking is a complete rescue of the CMAP amplitude, which for us, it's a very meaningful result because that seems to be, again, the driver of functional impairment in patients. and then also reduction in the number of hypomyelinated fibers. And this was achieved with a 3 monthly intravenous treatments. And then lastly, the mRNA, the microRNA approach. This showed a very, very robust behavioral rescue on all of those different metrics: rotating rod, grip strength, hang test, and hind limb strength. Again, about a 85% rescue of the motor nerve conduction velocity, and the CMAP in this case was just recovered to about 50%, but from very, very low amplitudes on the untreated mouse. And also very interesting, they did show recovery of some of the biomarkers that they were looking for. So neurofilament is a major one for linked to axonal degeneration. And I don't know if you can see my marker here, but so there was a significant decrease of neurofilament in the treated mouse, and also of another biomarker, in this case of the stress response. So overall, again, a very interesting and marked phenotypical rescue with all of those approaches. And the summary for CMT1A is that we now have several strategies that have passed this preclinical testing, including ASO, siRNA, and microRNA. They all lead to some degree of significant behavioral, electrophysiological, and pathological rescue. There was really no obvious safety concerns in terms of side effects on the mouse studies, and this has led DTX Pharma, the company that developed the siRNA, to be bought by Novartis, and they are now on track to start their first in-human trial for their approach in CMT1A. Now, moving to autosomal recessive CMT, those are CMT forms where we have loss of function, either homozygous or heterocompound loss of function mutations. And for those types of CMT, gene replacement therapies are expected to be the best strategy. So there are now several preclinical work in different types of CMT, including CMT1X, which is technically an X-linked disease, but where most mutations that have been identified are expected to cause loss of function of connexin 32, then CMT4C, CMT4J, and CMT2S, SMART1, or SMA with respiratory distress type 1. So all of those forms of CMT have been modeled in mice and have been tested with a gene replacement therapy approach. Interesting, most of those studies have been looking to both pre- and post-symptom onset treatment, which is, I think, a very important question that we're going to discuss again in the challenges. They all use the AAV9 vector in their latest kind of interaction. They did try an antiviral delivery initially, but now all of them transitioned to AAV9 after SMA success story. And then they all demonstrated some degree of improved muscle strength, increased motor conduction velocities, improving myelination, and other pathological features. This has led to now a phase 1/2 trial for CMT2S SMART-D1, which I think it's just starting to recruit patients. And then there's another story that I'm just going to briefly mention because it does answer a few of our questions, which is the phase 1 study that has been recently published of a gene replacement therapy for giant axonal neuropathy, which is a very severe complex neuropathy caused by mutations in GAN, and it's also a recessive disorder, so gene replacement therapy should be a good strategy. So here those results were published in the New England Journal, I think the beginning of this year. It's a little bit of a complicated analysis because it's a very small cohort, but they did make the case that there was some improvement in some of their functional metrics. But I think what's really most striking and really meaningful is that they could find in several of their patients either improvement or actually a resurgence of sensory nerve action potential responses after treatment. So suggesting that there was some meaningful functional results or from the treatment. The other point that they make, and I think it's also going to be a point that we're going to discuss again, is regarding the risk of the treatment. So all patients had some degree of adverse events. I think 100% had severe adverse events, and this is expected from the disease itself, but also from the gene replacement therapy. And that will raise the point of GAN is a severe disease. Patients will not survive past adulthood, but for the other types of CMT, that's not the case, and that risk-benefit balance may be a little bit different. In my lab, we're working on a specific autosomal recessive disease called CMT4B3, which is caused by mutations in the SBF1 or MTMR5 gene. It is a large gene, so just like Duchenne, we are developing this kind of mini gene approach to try to develop a therapy for those patients. CMT4B3 is ultra rare. It's poorly understood from a biological perspective, but very severe genetic neuropathy, again caused by recessive mutations on SBF1. And our approach has been to develop a mini gene that would fit a AAV delivery system. So the cDNA for MTMR5 is about 5.8 kilobases, so it's exceeding over 1 kilobase the cargo limit of AAV. So in a collaboration with the Zuckner lab and Matt Denzi, their computational biologist, we developed a couple of candidates that maintain the functional domains of SBF1, and we started now testing them in patient cells, and this is just a couple of examples of the delivery of the transgene. Two candidates, so candidate 2 and candidate 6 here. This is the wild type SBF1, candidate 2, candidate 6. In green is MTMR5 or SBF1, and MTMR2 is in red. Those are two proteins that are associate and colocalize. And we're just showing here that candidate 6 shows a way more homogeneous distribution than candidate 2. And then when we looked into immunoprecipitation, we could show that candidate 6 was being immunoprecipitated with MTMR2, so showing that they were associating. And we're now kind of moving to testing them into our iPS-derived motor neurons. Now, moving to, let's see how I'm doing on time. Okay, I think we're good. Moving to autosomal dominant CMT with gain-of-function mutations, there are two approaches that are now being developed. One is looking to strategies to specifically knock down the mutant allele, and that's particularly feasible in genes that tolerate haploinsufficiency, so meaning you can get away with just having half your gene expression, and I'm going to show an example of that. And then for genes that are not haplosufficient, using a knockdown replace therapy where you do two genetic manipulations. First, you knock down your endogenous expression, and then you replace with a gene replacement therapy approach. This could be another way to treat those gain-of-function mutations. So a little more complicated, but that's another direction. So the first work that I found for allele-specific treatments in CMT has been by Rob Burgess' group at Jackson Lab, looking to CMT2D caused by GARS mutations, where they use an allele-specific RNA interference to treat their mouse model, and actually they did it in 2 different mouse models of CMT2D, and they show a very significant rescue of the phenotype, both behavioral, electrophysiologically, and pathologically. If they treated the mice at birth, that was almost a complete rescue, and the more they delayed treatment, the less efficient the treatment was, which is kind of expected, but it's a nice confirmation of that. Then the next work has been done, actually a collaboration between my lab and the Gladstone lab that is developing this approach of allele-specific gene editing with CRISPR, and this was done for CMT2E, which is caused by mutations of neurofilament light chain. And they showed that they could, if they promoted allele-specific knockdown of the mutant NFL allele, they could essentially prevent the formation of some of the cellular phenotypes associated with CMT2E, namely the accumulation of neurofilament in the cell body of the mutant neurons, and also the release of neurofilament from the breakdown of axons in the motor neuron cultures. We kind of took the same approach, but now trying a more readily kind of translatable approach using ASO to mediate the same allele-specific knockdown. And this is work from Jessica Medina from my lab. And we use this model of human neurospheres from patients with CMT2E. This is a way to actually analyze axons in a more kind of direct way. And what she did was to design ASOs using essentially the same chemistry as nusinersen and tofersen, just to kind of have something that could readily translate to clinic. and she designed sequences that will target preferentially the mutant allele of NFL. She treated our neurospheres, and then they showed that she could see some improvement, though not statistically significant, in the amount of accumulation of neurofilament in the cell bodies of those neurons. But more importantly, she showed actually complete reversal of the neurofilament and peripherin increase that is seen in this motor neuron culture. So suggesting that the therapy could be beneficial, and we're now moving to animal studies for that. Now, the last 4 minutes, I want to discuss a little bit the challenges for genetic therapies in CMT, and I classify them in those 4 groups. And I think, again, there's a common theme with SMA, Duchenne, and other neuromuscular genetic diseases. So timing of intervention, this is specific to CMT, the delivery to peripheral nerves, the risk-benefit balance, and then clinical trial design and regulatory questions. So the first question is, how late is too late to treat patients with CMT? And as I mentioned, from at least the animal studies, it seems like even if you treat the patient after the onset of symptoms, you could still have beneficial results. Most of the beneficial results that we see are connected with demyelinating features, so we see conduction velocities improving even if we treat patients late. But the axonal features and behavioral features are not as efficiently treated at later time points. The first question is whether this will translate in human studies. And it could be the case that for CMT1A, it may be still very beneficial to treat patients even in adulthood, but for some of the axonal forms, there may be a limit to how much they will respond to treatment. And then if the efficacy is not there, would the risks of treatment be justifiable or not? Now, the other question is, can the different delivery strategies, either intrathecally or intravenously, reach the distal peripheral nervous system where the disease is most active? And this is work that hasn't been published yet, but was shared by Dr. Kleopas Kleopa and Armatus Bio, showing that, yes, if you deliver the transgene in non-human primates, you can actually detect the viral particles in the distal peripheral nervous system at 6 weeks and then 12 weeks post-treatment. Not only that, but you can actually see the downregulation of PMP22 happening both in the proximal as well as the distal sciatic nerve. So that was a good indication that, yes, you can accomplish delivery with a siRNA, with a AAV9 vector delivering the microRNA to the distal axons. There was another study that I mentioned before, the GAN giant axonal neuropathy study looking into the AAV9 delivery, and they have this unfortunate case of one of the patients passing 8 months after treatment from an unrelated post-surgical complication, but they had access to the post-mortem tissue, and they actually could show some distribution of their vector. But in this case was one of the patients that received the lowest dose of AAV9, so probably not necessarily what we would expect for all patients, but in this case they could not see any viral vector in the radial nerve, so suggesting that, of course, there's a dose dependency to that delivery of a virus to the peripheral nervous system. Now, another question, and that question I think it's very acute for CMT more than for SMA and Duchenne, where you have death as one of your outcomes. For CMT, at least for most CMT types, death is not an issue, so survival is not affected. Respiratory is very unusually affected. So patients will have a normal lifespan, they will have functional impairments, but would the risk of genetic therapy justify the potential benefits that they could offer? And I think that's a very active discussion. We know that even AAV9 having a great track record in clinic with SMA and with preclinical studies in CMT, the risk is not zero. There are serious side effects that have been reported in the SMA trials and since the approval of Zolgensma. So there's a very active question in the CMT community of what the best strategy should be, and at least what the first strategy should be for treating CMT1A. And of course, we have to take into consideration the patient's preference and willingness to receive a genetic treatment. And lastly, I'm not going to dwell too much on this because we're going to see this in later talks, but the question of how to measure treatment response in a slowly progressive disease like CMT is an active issue in the field. If genetic therapies provide a kind of home run result or improvement in patients, this will not be that much of an issue. But if we need to show stabilization of disease, we probably will never be able to do that very efficiently in CMT because of the slowly progressive nature of the changes that happen in the patients. All right, so my take-home messages are genetic therapy development for CMT is challenging but urgently needed. New disease-modifying therapies are now available for several ultra-rare neuromuscular genetic disorders, including ASOs, RNA interference, gene replacement therapy. Preclinical data support efficacy and safety of these approaches for several types of CMT, and clinical trials from some of those strategies are ongoing or about to start. And lastly, careful selection of patients and strategies necessary to optimize the benefits and reduce the risk and costs involved with therapy. And that's our group in Miami, and thank you for your attention.
Thank you, Mario. We have time for one question.
Hi, great talk. My question for you is partly related to CMT, but also for all of the RNA interference knockdown approaches more generally. But I think CMT 1A is particularly instructive. Would you worry? I would worry a little bit about excessive knockdown leading to the phenotype we don't want, which is HNPP.
Yeah.
And even for things like DUX4, do we know, is DUX4 essential? Do we know from animal models what a complete genetic knockout of DUX4 looks like? Why did we evolve to have DUX4? So those are some of my questions.
That's a great question. I didn't have time to really kind of approach the HNPP issue, but it is an active question, and a lot of the discussions on trial design is how to screen for HNPP symptoms. There are two problems. One is preclinically it's hard to induce HNPP in a mouse, so we don't know if that level of knockdown could potentially translate into issues for patients later on. As far as I know, in non-human primates, at least the microRNA approach has not yielded any kind of sign of HNPP, but it is, again, a very big concern for specifically PMP22, where we know there is a haploinsufficiency phenotype as well as the duplication. For DUX4, I don't know if Scott wants to chime in, but
So first on the HNPP, so the monkey data that Mario referenced, actually that's part of, I worked on that too, so that's part of our company. We don't see, the levels of knockdown we're seeing in peripheral nerve and Schwann cells are what we would consider to be in a therapeutically relevant level. So we don't see enough knockdown to create HNPP to begin with when we're starting at 150%. So that's one thing. And DUX4, it's early embryogenesis. It's probably found in the skin and maybe the thymus too. There's some evidence in that, but we don't know what it's doing there. So, but in skeletal muscle, there's no normal function.
Okay, thank you. We're going to move on to our final speaker for this session. So we have Dr. Heian Zhu. Thank you. So Heian comes from University College London, and today she'll be speaking about neuromuscular genetic therapies in COL6 and HSN1.
Thank you very much. Thanks, Vino, for the invitation, and thanks everyone to be here. And I'm very excited to share this work with everyone. So I'm a scientist, and all the work is from the lab-based research work, and would like to share with everyone and get feedback for our research work and to see how this can be translated to clinical practice. So first, I'll start with a summary slide about the ASO and the nucleic acid therapy, the drug approval from FDA in the last many years. Since the concept of antisense therapy started in 1978, you see how many FDA-approved drug, ASO and siRNA, so that's 16. And among those quite a lot as ASO drug at the beginning, and then they mainly focus on two mechanisms of action, that's splice switching antisense and also gene silencing antisense. And in the last few years, with the new chemistry and modification and technology developed, siRNA now also joins the market. So that's, I highlight here the different, I can use this, right? Here, okay, yeah. So I highlight in different color, you will see the different mechanisms of action, these drugs, and lots of drugs actually you see in neuromuscular disorders. There's 4 drugs for Duchenne muscular dystrophy and 1 for SMA, nusinersen, and also the very recent one for the tofersen for ALS. So this is the summary of this field. And also today we have heard very exciting talk about the FSHD, myotonic dystrophy, CMT. So what I'm going to talk about is focus on collagen 6. And so I will back to muscle and then back to neuropathy. So this is to understand this work. So it's very brief about the mechanism of action. Actually, I think I might be able to skip this slide because we already have a lot about the mechanism of action. So gene silencing, we have generic silencing and allele-specific silencing and splice switching. And also another mechanism is we call it's called TANGO, targeted gene upregulation. So that's something new, but still based on exon skipping or gene silencing, for example, that's used in Angelman syndrome and Dravet syndrome. So I will skip that slide, and then I'll start with collagen 6 muscular dystrophy. That's muscle one. I will focus on 2 strategies, allele-specific silencing using siRNA and antisense, and another one is exon skipping for a pseudoexon, and then move to SPTLC1 related sensory neuropathy. So for collagen 6 related congenital muscular dystrophy, this is the most common congenital muscular dystrophy caused by mutations in collagen 6, A1, A2, or A3. So all these 3 genes need to work together. Mutation in one gene will affect another 2, so will affect the function of the complex of collagen 6. So in clinic, there's a wide clinical spectrum covered from the mild phenotype, clinical phenotype, to the severe one. So for gene mutation, the severe type called Ullrich muscular dystrophy, and more than 50% of the mutation of the patients are caused by dominant mutation, and another less than half by recessive mutation. But for the milder basal myopathy, almost 95% patients are caused by dominant mutation. Why I want to mention this is the majority of collagen 6 congenital muscular dystrophy are caused by dominant mutation. The reason I want to mention this is because haploinsufficiency, that's something I want to talk about. So that's the paper published by Regan Foley from NIH many years ago. So in that paper, they present a case. So one patient is recessive, carry recessive collagen 6 mutation on one allele from one of the one parental allele, and carries a large deletion of collagen 6 A2. You can see collagen 6 A2, a large genomic deletion, and that's from one of the parents. And another allele, it carries a even larger genomic deletion, affect collagen A2. So A1 and A1, A2, both deleted. So, but the parents, they are the carrier, heterozygous carrier of this mutation. They are fine. There's no clinical symptom. So that's the rationale for our design. Suggests haploinsufficiency is not pathogenic. So allele-specific silencing to target the mutant transcript is possible as therapeutic strategy. So based on that, we have analyzed the genomic data in the Dubrovik Neuromuscular Center. So this is a summary of all the mutations we have. And you can see, so most of we mainly focus on the dominant mutation. Actually, you found lots of dominant mutations that are recurrent. So it's not just a single mutation. Many mutations that happen in many patients. So that also suggests our strategy to develop this mutation-specific therapy that can cover not just single patient, but a group of patients. So, and also, almost half of these mutations, they affect the spike cell, most of them call this in-frame deletion. So that's based on this, in the last 10 years, a lot of publication, not just from our own group, and also from other group developing antisense, mainly allele-specific silencing for collagen 6 CMD. The first is 2012 from Alessandra Fellini's group. They developed antisense target a SNP because this induces allele-specific silencing, and then followed by that 2 siRNA paper target. One is a spike mutation, another is a point mutation. One from H. Carsten-Bonneman's group, another from Noguchi from Japan. So they report that using SI to induce allele-specific silencing. And something new is in Noguchi's publication, they also mentioned introducing a mismatch to improve the allele-specific silencing. So for our group, we focus on we bring in the gapmer antisense. So before it's just the siRNA. So we designed the gapmer. The question we answered is we want to know if gapmer will preferably target pre-messenger RNA or mRNA, because we have a lot of splice mutation. The target sequence at pre-mRNA level and mRNA levels are quite different. So we want to know which target we should focus on. So that paper also mentioned, so by targeting mRNA is more efficient than targeting pre-messenger RNA. And then this is a very late publication from NIH using siRNA target another missense mutation. So almost this siRNA and antisense, this mutation-specific strategy cover almost more than 60% of the patient. And very recently, to improve the allele-specific silencing, we also develop, focusing on ASO strategy, we also report develop this study. So first we introduced the design of mixmer antisense, not a gapmer antisense. So mixmer antisense, the design is based on the secondary structure of our target sequence. So we check the difference between the wild type allele, one type sequence, and then mutant sequence. Found the difference, and based on that structural difference, we designed the multimer antisense. That design can increase the specificity to the mutant allele silencing up to 3-fold. And after that, we then introduce mismatch, called this mismatch mixmer antisense. By introducing mismatch to mixmer, we can further increase the specificity by 10-fold. So that's we published the paper in Molecular Therapy just a few months ago. So if you're interested, you can have a look and lots all the detail there. And also this paper suggests not just chemical modification, and structural modification is also quite important in antisense design. After the allele-specific silencing ASO, we also have another, this is something quite, one strategy seems quite promising for collagen 6 CMD. That is a mutation-specific strategy focusing on a very common recurrent DP intron mutation in intron 11. So that in a group of patients, around globally about 40 to 50 patients, we identified carrying that mutation. That DP intron mutation affects the splicing of intron 11, caused part of intron 11 being retained and included in the transcript. So you can see this, the top band here, this pseudoexon band here. So what we did, we designed antisense to induce pseudoexon skipping and to get rid of the extra intron 11 and make it back to normal transcript called pseudo-exon skipping. So that mutation, we have two different, we already complete this study, so testing two different chemistry. One is PMO design, another is MOE. So both strategies showed very efficient when we test in patients' fibroblasts. So we've done complete this work with the NIH team, testing PMO, and also the MOE, using MOE chemistry test in patients' fibroblasts. So we confirm at RNA level and also at protein level, this design worked very efficiently. So it's very promising. But then come to apart from silencing, allele-specific silencing, exon skipping, another strategy also been reported is using gene editing to silence, still for allele-specific silencing. So that's, we didn't know, this is from Carsten and also Carsten from NIH and Cecilia's work from Spain. So what they did for the deep intron limitation, they designed to cut that pseudo-exon, and it worked quite well in patients' fibroblasts. Another one is to silence, and Cecilia's group, they did is using gene editing and to silence that point mutation. And interesting thing is, as we know, CRISPR, there's adverse effect is off-target. Actually, when they found, yes, off-target is quite strong in that study, but the majority of off-target cause allele-specific silencing, and that's exactly what we need. So at this moment, these two studies still at a very early stage and still, low efficiency and off-target, undesirable off-target, still some issue need to sort out. So we still think the pseudo-exon skipping, that strategy is most promising for that particular deep intron limitation. And then ASO or siRNA allele-specific silencing is also has great potential for the dominant collagen 6 CMD. So regarding delivery, we already heard from previous talk from Charles about the challenge in delivery. For this condition, the same. So now we have very promising strategy, and then how to move this to clinical application. Delivery is the issue. Here is, as we know, without any conjugation, ASO, once we do the systemic deletion, almost 50% of ASO or siRNA will go to liver. And then another 40-50 will be cleaned by kidney. So think about, but after this, almost nothing left, maybe less than 10% of ASO can go to other body. And consider muscle as the biggest organ in our body, so very little will go to muscle. In that case, we do need to improve the delivery of for ASO treatment in neuromuscular disorders. For collagen 6, it's very special. Here, the target organ is not the muscle, as we know. The target organ actually is interstitial muscle fibroblasts. So it's a small cell population sitting between muscle fiber. To let those cell type uptake ASO, siRNA is quite challenging. That's what we are doing now as the next step. So we kind of move from target selection, now move to cell-specific delivery. So we are developing the fibroblast-specific delivery using peptide. We also open for collaboration. For example, as we know, the transferrin receptor antibodies seems quite promising for many muscular condition. So, and also we're looking for further industry collaboration for this. Now I'm going to move to sensory neuropathy. So that part of what has been published here. So The gene or mutation we focus on is the UK founder mutation, C133W. So this is from the work we've done in collaboration with Professor Mary Riley and Professor Francesco Montoni from Queen's Square Institute of Neurology. And we focus on UK founder mutation. At this moment, we have about around 70 adult patients affected by this mutation. They already have symptoms, and also we have around over 200 children carrying this mutation, although they haven't got disease onset yet. So we designed, to design the approach, we need to understand the mechanism. So for this mutation, the mutation in SPTLC1 affects the enzymes, is serine palmitoyltransferase. So mutation in SPTLC1 changes the enzyme substrate. So in wild-type SPT, it metabolizes serine is substrate, but mutant SPT, it changes substrate from serine to alanine, and then create a new metabolite called the deoxysphingolene or sphingosine. Those new metabolites cannot be cleaned out by our body, and then the accumulation of this abnormal metabolite will cause neurotoxicity. That's the reason we thought we will develop. We want to get rid of this toxic product. How to do it is design allele-specific silencing ASO or siRNA, particularly just get rid of the mutant transcript, and we keep the wild-type transcript. And also we know in clinic from animal model or other different type of from clinical mutation database, we know haploinsufficiency is not pathogenic in this condition. That's the reason why we developed this allele-specific ASO siRNA silencing approach for this condition. So we already developed, screened tons of ASO siRNA for the UK founder mutation, but there's a lack of suitable mouse model, animal model. All the currently existing 2 mouse models, they don't have human gene because this is a mutation specific. It's really target human gene. So that the current 2 existing mouse models are not suitable. They have a mouse sequence, not very a match with human sequence. And also we'll be concerned if this hypothesis will it work or not? And we do need to do the proof of concept study. So we talk about animal model, and very lucky we heard from Mary that David Bennett from Oxford, they have another mouse model, which is heterozygous S331F carrying that mutation. It's a severe mutation, and it's heterozygous. So it's a missense mutation. So we then got this mice from David, and we start the proof of concept study. And we first, I emailed David, can we get some fibroblasts? And then David said, no, you not just fibroblast, you can get the whole mice. So we have the mice sitting in our lab. So we designed ASO, test in skin fibroblast, just targets the mutant allele, and it worked quite well. You can see the green one is wild-type transcript, and the red one is a mutant transcript. In vitro, it worked beautifully, specifically silence the mutant transcript. So we select the lead and then test in mice. So we have 2 groups, as a neonatal group and adult group. And so we, in just single subcutaneous injection, and 7 days later we collect tissue. And you can see beautiful allele-specific silencing target, the mutant transcript. So this is that's mutant transcript. You can see the strong silencing in DRG and also in liver, so in neonatal group and in adult group. And there's no effect on the wild-type transcript, no silencing, at least no silencing on the wild-type transcript. So it's 1 week after, it's very promising. And then we will think, we need to repeat the study. So we do the repeat weekly injection. And then during that study, we're thinking, we know we want to get rid of the toxic deoxysphingolipid. And the liver is the major organ for sphingolipid metabolism. So similar like HTTR-associated amyloidosis, where they target, the major organ they target is liver. How about here? So, and then we design another group ASO using GANA conjugation, just enhance the drug uptake in liver in hepatocyte. So what we did, we repeat the injection weekly, and we also collect blood from baseline, and then 4 weeks after, 8 weeks after. So at RNA level, and then after 8 weeks injection, we collect DRG, liver, and the sciatic nerve. And you can see, so here we showed the liver, DRG, and the sciatic nerve. And you can see after 8 weeks, weekly subcutaneous injection in liver, more than 90% downregulation of the mutant transcript, and no significant change on the wild-type transcript, that's in liver. And in DRG, you can also see around 40% downregulation on mutant transcript, no effect on the wild-type transcript, and GANA conjugated antisense also further downregulate the mutant transcript in DRG. And sciatic nerve, it still show it seems quite challenging using antisense to target sciatic nerve. So this is at RNA level. Definitely liver is a perfect target. Oh, yeah, I'll finish very soon. So that's the weekly repeat injection. We got very nice data at RNA level. And then how about functional level? As you know, this mouse model, they don't show any neural behavior abnormality. This looks quite normal, so we do need a reliable clinical biomarker. Here, what we did is we focus on the blood level of doxycycline, DoxSA, the toxic product. And we monitor the blood level of DoxSA. So you can see this is in untreated mice. There's a wild-type control, and this age-matched the disease mice, dramatic increase blood level of DoxSA, and it looks very, very nice clinical biomarker. So we measure, and then we measure the blood level of DoxSA, and you can see in here, after 4 weeks, it's reduced, especially in the GANA conjugated group. And after 8 weeks treatment, it reduced further, especially in the GANA group as well. So this suggests GANA group works much better than unconjugated group. So just to summarize, in collagen 6 congenital muscular dystrophy, and we think RNA-based therapy is a very promising approach for this condition, and there's quite a lot of strategy we can use, allele-specific silencing, exon skipping, and gene editing. And then, but still, there are lots of question and challenge we need to overcome, especially the delivery. And for SPTLC1 sensory neuropathy, definitely allele-specific silencing, just like Mario talked about the CMT, is very promising strategy. We have already completed the proof of concept, and a new suggestion is targeting liver will be more efficient than targeting other organs. And also, from the blood level of DoxSA study, you will see actually the mice show increased tox product from very early stage, 7 days old. That suggests, do we need to treat patients at very early stage? Means those biomarkers actually already become abnormal in the very early stage. Do we still need to wait for the symptom onset, or we can treat patient at the pre-symptomatic stage? I think that Mario also talked about this in the CMT talk. So that's also happening as PTLC1HS. So I think I will finish here, and I would like to thank my team and also Professor Mary Riley and Professor Francesco Montoni for all the advice for this project. Thank you very much.
Thanks, Ayaan, for a great talk. We're running a bit late, so I think it would be great if people could ask questions to you over coffee.
Yeah, happy to take a question during coffee break. Thank you.
And so now we're going to a short coffee break.
So we're going to get started now, and this is session 2, which is going to include flash presentations of about 10 minutes length of various topics in neuromuscular disease. So our first speaker is Katie Dodd, and she's from Manchester Center for Clinical Neurosciences. And the title of her talk is Refractory Myasthenia Gravis, Characterized by Widespread Innate and Adaptive Immune System Changes.
Great, thanks very much. So yeah, my name is Katie Dodd. I work in the myasthenia clinic in Manchester with John Sussman, and I'm going to present some work that I did as part of my PhD at the University of Manchester, working with Madhvi Menon at the Lydia Becker Institute. And I'm really grateful to the Neuromuscular Study Group for funding some of this work through the Shark Tank a couple of years ago. So we're all quite familiar with the pathophysiology of myasthenia. We know that there's pathology within the thymus, and this leads to autoreactive T and B cells, which generate pathogenic autoantibodies, and AChR antibodies activate the complement system, and then the antibodies and complement both lead to damage and problems with transmission at the neuromuscular junction. And it's quite an exciting time to work in myasthenia. There's been this explosion of new therapies. But what we're really lacking is biomarkers to help guide treatment decisions. And so now with these new avenues for treatment, what we're going to need is biomarkers to help us decide who's going to have severe disease or how to try and decide which therapy to target to an individual. And so we looked at 20 controls and 58 patients with AChR positive myasthenia, recruited into 4 quite specific groups. So a group that were stable on no immunosuppression, they just needed a little bit of pyridostigmine. A group who'd been given azathioprine or mycophenolate and stabilized. A group of refractory patients who'd had multiple treatments and failed to respond, and some treatment-naive patients. And I've looked at their immune profile, mainly using flow cytometry on peripheral blood mononuclear cells. So we know that the antibody titer doesn't well correlate to disease severity, and that panned out here. What we sometimes use to try and monitor azathioprine or mycophenolate is the lymphocyte count, but we didn't really see that there was a definite difference between the different cohorts with that. Looking at the B cell frequency, you see the CD19 count is lower in those on immunosuppression, but doesn't effectively distinguish between those who had responded and those refractory patients. And so you can look in more detail at the B cells, looking at various different markers on the cell surface. So looking at CD24 and CD38, we looked at memory B cells, and you can see that these are expanded in disease compared to control. And this was mainly in those refractory patients. Now, interestingly, this also, when you split between those with early onset and late onset, was mainly in those with early onset disease, and that might translate to differences in response to memory B cell targeting therapies. The B cells, when stimulated, also demonstrated a pro-inflammatory response with higher production of IL-6 and TNF-alpha in disease compared to control, but mainly in those refractory patients. And looking at the myeloid system, we see this pro-inflammatory feature there as well, with an expansion of monocytes in disease, mainly in those disease-active groups, the refractory and the treatment-naive, with ongoing symptoms. But we also saw that there's a loss of dendritic cells, which can promote an immune tolerance in refractory patients. One way that dendritic cells promote immune tolerance is to promote Treg differentiation. And when we looked in the T cell compartment, we can see the Tregs are lost in those refractory patients, and this correlated with disease severity and quality of life scores. But it also correlated with those pro-inflammatory B cells. The pro-inflammatory B cells limit Treg differentiation as well. And this is another potential avenue of therapies that perhaps isn't being as well explored at the moment. There's various different ways you can try and promote Treg development, which might help patients with refractory disease. Looking at the complement proteins, we found that C3, C5, and clusterin were all higher in those refractory patients, and again, these correlated with quality of life and disease severity scores. And complement doesn't just interact with the neuromuscular junction, it also interacts with the lymphocytes, and here you can see a complement receptor on CD8 T lymphocytes showing higher expression in refractory patients. Of the 10 refractory patients, 8 of these went on to have CD20 depletion with rituximab, with variable response, but 3 I've classed as non-responders. They had a less than 50% reduction in their MG composite score, and they're in red here. And you can see that these non-responders had a low, less than 3% B-cell frequency at their baseline. And what they did have was evidence of complement activation, so sort of persistent high levels of complement or complement receptors on the lymphocytes throughout, and therefore these patients perhaps would have been better suited to a complement inhibitor therapy. We know that B-cell depletion promotes Treg development, and that again fits with the pro-inflammatory B-cells. You get rid of them and the Tregs are allowed to differentiate. And what we can see is that although you deplete the B-cells, there are some B-cells left, and they are mainly plasmablasts, and that just shows the problem we have with CD20 therapy, in that we know that the B-cells, as they differentiate, become antibody-producing cells, they lose their CD20 expression, and so it's just highlighting this, the reason that not everyone responds to rituximab. So in summary, I've had to kind of briefly whiz you through some various biomarkers that might, with a bit of further validation, be useful to help guide treatment decisions, and some various thoughts on the treatment implications of these. I'd really like to thank the people at the University of Manchester, some help from Cardiff University, but also the clinical collaborators I had from around England to get some of these patient samples. So thank you.
Hi there, thank you for a great and brief talk. I think one question that comes up right away for me with refractory myasthenia early onset is gender differences. So was this looked at? I know you only had, or not only, but you had 10 refractory patients, but were gender differences looked at?
Yeah, it's tricky because of the sample size, really, and also the fact that most of the early onset were female and most of the late onset were male. On that graph, they were differentiated into male and female, and there wasn't a clear gender difference. And yeah, if you do split them out gender-wise, it doesn't, but I don't think I had quite enough numbers to say that for certain. Tregs as well. There wasn't a difference between early onset and late onset, yeah.
I have one question. What are your future?
So, I mean, I think this needs further validation. What would be good is to do a prospective study and pick up patients as they're diagnosed, and then find out if that does actually correlate with how they do further down the line. Thanks very much.
All right, well, for our second speaker, we have Michaela Nani, and she is going to be presenting on remote monitoring to improve adherence to physical exercise, a pilot experience at the NEMO site.
Good morning, and thank you for the opportunity to talk about our project. Maintaining an healthy lifestyle with a regular motor activity is difficult for everyone. And this is observed in patients with DM1 because they have more difficulty, because they have avoidant and passive behavior, and also they have already fatigue and weakness. The aim of this study was to verify whether remote monitoring could improve adherence of physical protocol exercise. This is a pilot study in which 15 adult patients with DM1 were subjected to in-person PT training. So they were divided into different groups. 6 of them were enrolled in unsupervised home-based program. So they have to complete physical exercise independently for 3 times per week. And 9 patients were enrolled in a weekly remote monitoring program for 12 months. So they have to complete physical exercise independently for 2 times per week, and remotely with a physiotherapist live for 1 time per week. Also, monitoring visits were scheduled at 3, 6, and 12 months by a multidisciplinary team, and routine neuromotor function tests were compared between the two groups. The protocol presents 3 different phases. First of all, there is in-person health education by a nurse, so we talk with the patient about the importance of the adherence in the physical exercise with the tracking modalities. This is for, This is for all the patients from both groups. The second phase is about the physician and physiotherapist assessment. So we create a rehabilitation program with a video that all the patients could refresh with the user QR code on the leaflet. And only for the telemonitoring group, there is a weekly monitoring of quantity and quality of exercises. These are 3 examples of the exercises that the patient could refresh with the QR code on the leaflet. And finally, there is the last phase about the assessment in 3, 6, and 12 months patient in which we want to investigate patient adherence and compliance, motor outcome measure, and also patient perception of burden with the use of MDI. Preliminary results from 3 months data show us that all the patients from the telemonitoring group adhered at 3 months follow-up visit, instead only 1 from the other group. And also remote group reported an improvement of motor outcome measure and also an improvement in perceived burden of activity and fatigue. So digital is already in neuromuscular disease, but remote monitoring have an added value for patients with DM1 because could help them to improve adherence in care recommendation. And also in the era of therapeutic intervention, we think that we could implement this type of assessment because may maximize the action of the treatment. Maybe with the help of a regular telemonitoring of physical exercise in trials. Finally, our future intention are to implement this program with nutritional assessment and biomarker analysis. And if you want, you could find this topic at this QR code in which there is my poster about BMI and insulin-resistant telemonitoring protocol. Thank you for your attention.
Thank you very much. That's very interesting. And obviously, engagement in these programs can be a barrier, can be a problem. Did you actually involve any of the patients or in a patient advisory group at any point to discuss actually what might work best for them so they can advise this process? Or is there a plan to do that afterwards? Because if we want behavior change, then involvement is actually really key.
Sorry?
Is there any plan for actually working in partnership with patients in terms of planning how they prefer to engage with these sorts of programs so that you can encourage behavior change for engagement?
Yes. Okay, thank you.
Sorry, yes, this is true. This is just a pilot thing. They are having a satisfaction and a free text space to let us know where we need to improve and which exercises are to be passed or blocked. So yes, we are working with them. And she's the research nurse who is doing this, so she just didn't understand the question.
All right. Thank you, Michaela.
Thank you.
Up next, we have Katie Lillig with trial of oxaloacetate in ALS and TOALS.
chair. I'll just watch out.
Perfect. Hi, everyone. My name is Katie Lillig. I am a senior coordinator from the University of Kansas Medical Center. I have worked on the trial of oxaloacetate in ALS, and I want to tell you guys a little bit about our findings. So first off, amyotrophic lateral sclerosis, as many of you know, is a progressive neurodegenerative disease, typically results in death approximately 2 to 5 years from the time of symptom onset. Mitochondrial dysfunction may play a critical role in ALS neurodegeneration, which has suggested an important therapeutic target in ALS. Oxaloacetate was safe, well-tolerated, engaged brain metabolism in patients with Alzheimer's disease. It activates mitochondrial bioenergetics, increase both respiratory glycolysis capacity, and has been shown to be cytoprotective in cell models. And you can see from our chart on the side that in mouse models, when injected with OAA, there was increased hang time translating to strength. So our specific aim was to determine safety and maximal tolerated dose of OAA in patients with ALS. We also had a sub-aim to look at the pharmacokinetic profile of OAA in ALS, and then lastly, to determine target engagement. And we did this through TDP-43, mitochondrial biomarkers, and MR spectroscopy. So a little bit about the study design. It was a prospective phase 1B, 3+3 design. It was a very brief trial. It was only 28 days. So we had screening, baseline. Once the patients began dosing, we would call them weekly for follow-up to check in on any adverse events. And then day 28, we would follow up with labs and final questionnaires. So dosing began at 1,000 milligrams oral twice daily. We increased cohorts by 500 milligrams up to 2,500 milligrams twice daily. We were looking for dose-limiting toxicities, which is defined as any serious adverse event related to OAA or any adverse event that required patients to stop taking the drug, or a twofold increase in ALT and AST. We ended up enrolling 19 subjects. We had 1 screen fail, and 1 patient did withdraw due to a dose-limiting toxicity. This patient was at the highest group, so we enrolled 3 more patients, but overall it was well tolerated. The PK data did not detect OAA in the absence of a positive control. For the small sample analyzed, target engagement did not show us a clear signal. Looking at our adverse event table, you can see as the dose increased, we did experience more adverse events. The most common adverse event was GI upset, heartburn, which was remedied through taking the medication with a meal and over-the-counter Tums. We had 1 serious adverse event, but it was not related to the drug. With TDP-43, you can see there's not a significant change from baseline to end of treatment. However, there is a noticeable declining trend in the OAA-treated patients, indicating a reduction in TDP-43 phosphorylation. We also looked at mitochondrial health index biomarkers by dose. The pre and post treatment indicate the low and high dose cohorts may have better target on mitochondrial function. However, we had a low number of patients in each cohort, which limits our conclusion. So overall, OAA was well tolerated up to and including the 2,500 milligrams twice daily. There was a positive signal with the MR spectroscopy through the small sample size analyzed. Other target engagement did not show a clear signal, and the PK data was not measurable, likely due to an issue with our detecting methodology. So future direction, a potential randomized placebo-controlled trial would be our next reasonable step to look further at efficacy and target engagement. And of course, we want to thank all of our patients, our study team, and Terra Biological, who very generously donated OAA to our trial. And thank you to Dr. Jaudat and Dr. Statlin for being my mentor. Thank you, guys.
All right, show of hands for any questions. Raise them high so we can see them.
I have a question, Katie.
Sure.
What type of physical function measures would you test in your prospective trial to try to test, like, for strength changes?
Yeah.
Or?
We typically have done hand grip strength or HHD just in about 4 or 5 different muscle groups, so I feel like we would most likely do that if we go to a bigger trial.
The reason why I ask is it seems like the mechanism is through, mitochondria, and those are like strength, and so maybe some type of endurance type of metric, or fatigue would add some benefit.
We actually did add on the last 6 patients fatigue severity scale. We haven't had the chance to get the analysis, but I think, fatigue would be a good measure to add.
We did not.
Sorry, that was what I was going to ask. Neurofilaments getting measured in ALS clinical trials, and it can provide some prelim evidence that the drug might be working, even with small numbers. So I was curious if you measured it.
Not with this small pilot study, which the main goal was to get a maximum tolerated dose, but yeah, it would be reasonable for future.
Other questions? Thank you so much, you guys.
All right, Brendan, who's next?
All right, we got Lawrence Hayward from UMass Med, and he's going to be speaking about outcome measures to quantify longitudinal changes in motor function in FHD.
All right, thank you very much. I'm Larry Hayward. I'm at UMass, and I'm excited to present some of our highlights from a small pilot study we're performing as part of our Wellstone Center for FSHD. These are my disclosures. I've been a site PI for the Fulcrum trials. And as we heard from Scott Harper earlier, FSHD has very intriguing and complicated genetic and epigenetic features. And so I'm not going to go into this further other than to just point out that I've also always been intrigued by the clinical variability and asymmetry of muscle involvement in FSHD. And I think there's still a lot we need to understand about what are the factors that are contributing to that. So what we've been doing as part of our Wellstone Center is setting up a small pilot project. This is literally just 10 FSHD patients and 5 controls to test some novel measures that haven't really been explored thoroughly so far, and could offer some potential avenues for gaining more high-resolution granular data on patients, not only in FSHD, but potentially in other diseases as well. So I'm going to focus on some of the things we've been looking at. There's more information in a poster that I'll have here tonight. These include some functional measures, including active workspace volume, which is a way of looking not only at just the shoulder joint, as in the reachable workspace, but in some other functions that can involve bending and paraspinal muscle involvement to track that with disease. And another measure that we've been looking at, which is very exciting, is called OpenCap. It's basically a way of using 2 cameras to ascertain 3D trajectories of the joints. That is a low-cost method, and I'll show you some data with that. We're also exploring the use of a 100-meter timed walk-run to see if this can be a useful measure of maximal ability in FSHD, for which it hasn't been applied yet. And then other areas in our Wellstone program, we're looking at plasma and urine biomarkers. I'm going to show you some of the muscle ultrasound imaging that we've been performing and correlating with muscle MRI. And we're comparing in this same patient cohort to some of the other standard measures that have been pioneered in the ongoing larger MOVE studies with Michaela Walker and Jeff Statland here, so that we could relate our observations to some of those patients potentially. So this is this just a summary of, it's a small number of patients, it's ongoing, we're doing 6-month visits and doing a range of some of these assessments. So this is just to show when we look at grayscale ultrasound and use qualitative assessment blinded by our radiologist comparing across visits, we are seeing, even at 6-month intervals, we are seeing changes in the muscle pathology visualized on that test. And potentially this could be used to follow patients over time. It's also been demonstrated in much more detail by the group from the Netherlands and other groups. This is the summary of some of the correlations that we've seen. So if we focus on the average HECMAT score of our ultrasound, we see a fairly good correlation with the Ricci score and Lamperti score. And we're also correlating this these various measures, including the RODS patient-reported outcome, as well as the measures of muscle strength. So I have more detail on the poster later tonight. I'd be glad to discuss with people. But this is promising in that, if we can use some modalities like ultrasound, which are fairly easy to do in the clinic, we can potentially gather more information at more frequent time scales than MRI and include more patients. So we are also doing MRI on this small group of patients, and it's really been interesting collaborating with Springbok Therapeutics, who are using AI-type approaches to segment individual muscles from the scans. And it's very interesting to see some of the patterns that are coming out, where we see, for instance, in a lot of the leg muscles, there's this distal to proximal gradient of fatty degeneration in the muscle. This has been observed before and reported by other people, and Springbok recently has a publication of a larger set of MRIs. But to me, what's really interesting is that, what does this suggest about the origin of the pathology? Is DUX4 somehow being epigenetically de-repressed earlier here and turning on and leading to degeneration? Or does this reflect more of a susceptibility of different regions of the muscle to the DUX4 expression? So these are unanswered questions that I think need to be looked at in more detail. This is the active workspace volume, which was developed by one of our collaborators in the Wellstone Center, Linda Lowe's at Nationwide Children's. And with this approach, it's using a 3D camera to, it was originally designed to evaluate boys with DMD, and it was designed as a video game, so it's very engaging. It keeps their attention, and basically the instructions are that they are, as long as they stay seated, they're allowed to bend side to side or forward and backward to reach their target, which represents a real sort of a more realistic functional outcome for someone who may be unable to get up from a chair but still needs to reach for something. And so we're trying to see if including the use of these muscles would also provide a benefit in tracking patients longitudinally. This is an example of OpenCap. So basically all that is needed are two iOS devices set up on tripods. And this was developed at Stanford in the bioengineering department. So essentially the two videos are synchronized and uploaded to a HIPAA-secure server. And then the parameters that are extracted include the position of every joint through space and time. And these are plugged into basically kinetics models. One of them is called OpenSim, but there are other models. Once you have the raw trajectory, you can use different models to analyze the data. And what we can see, an example of some of the parameters we can extract are these various, we can get the knee angle, the hip flexion angle, the lumbar flexion angle, and we can plot that over time during a given activity. So this is sit to stand times 5, a standard test that's done where normally all we get is the time that it took the patient to complete the maneuver. But here we get much more granular information about how the joints are working in tandem that could give us some ideas about changes in control of the muscles. And if you notice in red here, there's another set of tracings, and these are the lumbar bending. So again, some patients with FSHD have trouble getting up from a chair, and depending on their distribution of weakness, they will frequently lean to one side or another as part of that maneuver. And so we're able to see some differences between FSHD and control, and this is something that potentially could be monitored again over time for the natural history or in response to a therapy to see if changes in some of these muscles that we don't normally look at might be affected by a treatment. And this is just an example of the rich data that we can obtain from this. These include repeated trials with fairly good agreement, and then some patients, such as this FSHD2 patient, that of necessity required a lot of leaning in order to get up from the chair. So I just want to, there's a lot more information in my poster. I would encourage anyone to stop by if you'd like. And just want to point out some of the other collaborators in our Wellstone Center include Linda Lowe's, as I mentioned, Russ Butterfield at Utah, Doris Leung at KKI, and Scott Harper. And so we're proposing to do a larger study of about 80 patients in our Wellstone Center to try to look at the most promising of these outcome measures and hopefully incorporate them into other natural history studies. Thank you.
Lawrence, are you doing any, so one thing that ultrasound could help with would be maybe, you could also look at facial muscles. Have you done any ultrasound of the facial muscles?
We haven't, but that's a very important area. I think that would be really interesting to, clearly that would be useful because there's been a much more appreciation recently of the importance of that.
And an MRI would be more challenging there.
Right. Yes.
That was very interesting, Larry. The changes along the course of a single muscle that you're showing on the MRI, how do you think about that in terms of, are those changes of signal within a single fiber, or does that reflect different populations of fibers, and how does it relate to kind of the pennation angles in those different muscles?
Exactly. So as we know, some muscles taper down to a tendon, and one could imagine the force concentrating at those tendons. Like in DMD, it seems like there's degeneration from both ends of a muscle when that's the case. That doesn't seem to be the case here, and it makes me think more about other characteristics of the physiology or the metabolism of the muscle. And so it's very interesting that if you look, for instance, there are studies in human tibialis anterior that show that after exercise, the recovery of the ATP synthesis and phosphocreatine occurs much more slowly in the distal end compared to the proximal end. And so this could relate to, the physiology itself, or something about the anatomy and the pennation and the way the muscle is attached. It could, I think it's something that needs to be explored more, because getting back to the earlier discussion about modifying factors, clearly there are genetic modifiers of FSHD. I mean, if you think about SMCHD1 and the epigenetic genes of known epigenetic function, those are pretty strong modifiers. There could be other genetic modifiers that affect the DUX4 activation or expression, but there could also be other physiological modifiers some of which could be genetic, that affect the susceptibility of different portions of the muscle. And it could even be an effect of the vascular supply. So there's a lot of questions that this opens up, because it's hard to imagine, one scenario would be you have DUX4 continuously being generated at the distal end and then somehow a wave of degeneration, but it seems a little less plausible than something like a differential susceptibility along the length of the muscle.
Alrighty. Thank you, Dr. Hayward. Who do we got next, Brendan?
All right, we got Georgia Corradi from Catholic University, and she's going to be speaking about treatment effects on ambulation, loss in spinal muscular atrophy type 3, insights from the Italian ISMAC registry.
Yeah, well, I'm not Georgia Corradi, obviously. She is sick and unfortunately with a bad COVID infection, so I'm presenting on her behalf and on behalf of the Italian SMA network. So the aims, sorry for the extra A, of this presentation is to see whether the disease-modifying treatments so far in SMA have changed the time of loss of ambulation. And how we did it, we looked at the data collection from the Italian SMA Consortium and Registry. We defined loss of ambulation pre-treatment, that is 1 year before treatment was started with the disease-modifying treatment, and post-year, as you can see. And the sample size is rather big. We have double that number, but this is the analysis from half of the sample. And as you can see, there's a different distribution of all the SMA types. We do have a number of patients who are treated. And as we expected, the treatment group has had a significantly lower risk of losing ambulation, as you can see by the graphs. And we did look at other confounding factors that some were quite expected, like the copy numbers. We looked at gender differences, whether type 3A or B, because we know they are very different, and whether the standards of care could actually change this. And the copy numbers did actually show that the patients, and they were not a small sample in our group, with more than 4 copies had a risk of losing ambulation before the expected time by 61 point, as you can see there, 0.7. And the gender effect had no effect on the analysis. 3B patients had a better chance of doing better with treatments. And the standards of care and how we looked at this, we decided to look at if the diagnosis was from 2017 onwards, which was the date, the year when the first drug approved for SMA came into the market, and then we compared it to the previous standards of care, which were back in 2007, and then we did actually see a difference from 2017 onwards. We didn't catch a difference from 2007 to 2017. That was stable. But I think, again, this shows that the moment the treatments came, the standards of care became more applicable, and sort of the patients were more adherent, and so were the centers. But we did find that there were additional things that we did not expect to see in the cohort, so that there were quite a number of patients who were actually still untreated when we started the analysis, and a number, as you see, which is quite significant, had more than 4 copies. So we also stratified patients by 3A and 3B, treated and untreated, because they do actually have a different time course, even in the natural history. And as you can see, there's a similar risk of reduction of loss of ambulation. And we did also stratify by the number of copies, and you can see the risk there of losing the ambulation. And so in conclusion, when we put those things together, we do think that, as we expected, treatment significantly impacts the time of losing ambulation, and it does act as a modifying factor with those other two expected factors being 3B and having more than 4 copies of SMN2. So I think this is important for counseling with families. We know we did expect some of these results, but when you have it there to show and talk about numbers, I think it's helping now to consult with families because of the 3 and more options that are coming along with SMA. Thank you.
Any questions?
Great talk, thank you. I'm actually dealing with this right now with a patient where we're not sure if they have 4 copies or 5 copies. And it sounds like the consensus is for 4 copies you would treat. And for your 4+ copy patients, did you distinguish between, sort of 4+, where it could be 4 or more, or actually knowing that they have 5 copies?
So we did have a technical concern with the number of copies coming out from the different labs throughout the country, and we did have a centralized look at all the copies because some were captured as fragments. So when we talk about actually 4 copies or more, we're quite confident that we're talking the right numbers. When we say 4 copies or more, it's really 4 copies or more. We did actually then stratify by 5. Few of them have even 6 or 7. Those we are not treating. But the 4 copies we are treating. Most of us are. So.
Any other questions?
Thank you.
All right, up next is Dr. Kelly Gwathmey, and she's presenting on Myasthenia Gravis Foundation of America Global MG Patient Registry, Characteristics, Insights, and Learnings After a Decade.
All right, good morning, everyone. I know I'm the last one standing between you and lunch, and so I'll try and make it brief. I just want to say thank you to the organizers for inviting me to give this presentation on behalf of my co-authors that have been working on analyzing the data from the MGFA Global MG Patient Registry over the last decade. I don't think I can advance. Here are my disclosures. Okay, the objectives are to briefly describe the MGFA Global MG Patient Registry and to report the baseline demographics and disease characteristics of this registry. And so, as many people in this room know, this is a self-reported survey of patients with myasthenia gravis, and it was established in 2011. The first participants were enrolled in 2013, and participants are surveyed twice a year in the summer and winter time. And so there are about 3,500 participants. I'm going to show you that data here momentarily. If we assume that there are 78,000, give or take, myasthenics in the United States, then that makes up about 4.5% of all the myasthenics in the US have enrolled in this registry. And to compare it to the type 1 diabetes registry, where we have 2 million type 1 diabetics in the United States, 14,000 have enrolled in that registry, making up 0.7% total. So we think we're doing pretty well. All right, so the MGFA Global MG Patient Registry is currently powered by Health Storyline. So that's under the Alira Health platform, and it moved over from UAB a couple of years ago. And so this patient registry characterizes the natural history of the disease and its impact on patients over time. The surveys are administered directly to the patients, and enrollment is open and longitudinal follow-up is open-ended, so you can stay in the registry for many, many years. So in terms of the design and methodology, you must be 18 years or older in order to enroll, and you have self-reported myasthenia gravis. And so we have descriptive analyses conducted on key clinical features and variables, which I'll be demonstrating shortly. And then enrolled subjects are contacted, as I said, twice a year to provide updates. And then we're going to be showing you the data from July 2013 to June 2023. So we have patient-reported data, again, age, gender, sex, race, ethnicity, state, weight, employment status, and income status. And then we also capture patient-reported outcome measures, including the MG-ADL and QOL-15R. We also are capturing clinical and treatment characteristics, including antibody status, medical comorbidities, exacerbations, thymus gland status, thymectomy history, and treatments overall, as well as healthcare utilization, resource utilization, including overnight hospitalizations, ICU visits, and emergency department visits. And so looking at where we're pulling the patients from, you'll see the dark kind of orange is where we have the most patients. And so I think it's the 328 is the maximum in Florida, then followed by Texas and California as the highest enrolling states. And so looking at the breakdown in terms of the demographics, we have nearly 40% men and just over 60% women, for a total of 3,556 in this decade's worth of data. And so nearly 95% are not Hispanic. And in terms of age at diagnosis, we see that, and again, just showing you the averages here, that the average male age is 58 at time of diagnosis, and female age is 43.9. And so, not surprisingly, some time passes and then the patients are enrolled. And so for the men, average age at time of enrollment is 62.9 years, and for females, 51.2. Looking at the antibody serostatus classification, I want to draw your attention to the fact that the sample size is much smaller here. It's just over 1,800. And the reason is we really filtered out the patients who did not know what their antibody status was. And so of those who clearly knew, 62.8% reported to be acetylcholine receptor antibody positive, 31.6% were seronegative, and just over 5% were MuSK positive, and 0.4% were LRP4 positive. Looking at employment status, I think this is extremely important. Almost everyone answered this question. So full-time employment, only 34%, and part-time employment was 9.9%, and 56.1% of the patients surveyed were unemployed. And looking at the age at diagnosis for the myasthenia patients stratified by sex, you'll see this really beautiful bimodal peak here. So the women are outpacing the men up until 51 to 60 years of age, and then at 61 to 70 years of age, the men start to outpace the women, as you would expect, and so that's really beautifully represented in this data. I'm sorry, this probably projects very small on your screen, but looking at the treatments and therapies taken by our myasthenia patients, remember that the data cut here is June 2023, so a lot of our newer therapeutics had not yet come to the market or had just recently come to market. And so that's why you will see, for example, only 7 patients reported taking Vyvgart and only 1 patient reported taking Ultomiris. The vast majority of patients had taken, not surprisingly, pyridostigmine and prednisone, followed by IVIG in about 1,500 patients. And then in terms of the nonsteroidal immunosuppressant therapies, we saw that mycophenolate followed by azathioprine were the most common. Looking at comorbidities, perhaps not surprisingly, hypertension, hyperlipidemia were the most common, but I really want to draw your attention to number 3 and number 4, which are depression and anxiety, which is quite concerning and perhaps part of my talk in a few moments. And then we also looked at other medical comorbidities that are autoimmune in nature, and so you'll see there was quite a high prevalence of autoimmune thyroid disease in 483 patients, for example, and then we also captured rheumatoid arthritis and lupus data as well. We also are able to look at the MG-ADL score and stratify it by sex as well as serostatus. And so you'll see in these 3,500 patients, more or less, the average MG-ADL score for the men was 5 and for women 7. And if you separate it out by antibody status, you'll see that the AChR positive patients had an ADL score average of 6 and LRP4 was 8.5, but please note that was only 8 patients. And then for the MuSK patients, it was an average of 7 and seronegative an average of 8. And so what are the take-home points? This is a decade's worth of MGFA global MG patient registry data describing baseline characteristics of over 3,500 MG patients. The age of diagnosis was stratified by sex and stereo status. Comorbidities and prior and current treatments were described. Employment status is also captured, as well as the distribution of the MG-ADL scores by sex and antibody status at time of enrollment. So these findings do support the value and ongoing utility of a large-scale patient-reported registry, such as the MGFA Global MG Patient Registry. And so the manuscript is in preparation and nearly complete, and hopefully to be submitted soon. And then if you want to learn more about the MGFA Global MG Patient Registry, there's the QR code. You can also find it on the myasthenia.org website. Thank you so much for your attention.
We have a question over here to start.
Lots of questions. Okay.
Hey, thank you for the talk. What was the basis of the diagnosis for the seronegative group, like single fiber EMG, slow RNS?
Right.
So their percentage is higher than what we normally see.
Right, and I think that's one of the downsides of a patient-reported registry information like this is the accuracy of the data. And so the way there was an algorithm to calculate out who was considered seronegative. And so if patients said they know that they were tested for those antibodies and they were negative, but told they have myasthenia gravis by their provider, then they were considered seronegative. We did not require, if I remember correctly, that they state they had a single fiber EMG for confirmation, but if their provider had diagnosed them with MG and their antibody testing was complete and negative, then we went ahead and concluded. Thank you.
Thank you. So I was struck by the number of patients who have peripheral neuropathy. You have over 700 patients. Did you look further into that, whether they had diabetic B12 deficiency or other causes?
Did we look further into the type of peripheral neuropathy or the cause? No, it was simply a question of whether or not you have peripheral neuropathy. We didn't link it back, and I don't believe have the ability, well, we might, to track back and see how many of those have diabetes and things like that. But no, we've not analyzed that specific degree of detail yet. Yeah, Mike.
Great talk. So for the antibody status, your acetylcholine receptor antibody level was like 60%. Did you stratify ocular? I have two questions, ocular versus generalized. And then my second question is, your ADL score is very high in the registry at 7 across all groups. And did you hypothesize why patients may be undertreated in the registry?
These are good questions. I don't know the stratification based off ocular versus generalized for the antibody test, but we would be able to analyze that. We have the data to do so. We have not done that yet, but that's an excellent point. I don't have that in the tables that we have prepared. In terms of the ADL score being very high, I mean, first I will say that's one of the limitations of a survey like this, is it sort of self-selects patients that are often a little bit sicker. And also, you remember this goes back to 2013, and so clearly our treatment landscape has evolved since then, and so hopefully, we'll see trends over time. That's one of the beauties of this type of longitudinal data, that the ADL scores will start to improve with the, implementation of these newer therapeutics. But yeah, I mean, I do suspect there is some undertreatment, especially going back before 2020 or so.
I was just wondering, from a regulatory issue, how'd you handle IRB and informed consent?
It is through a, I'll have to get the IRB data for you. We have it is IRB approved. I can't remember where that specifically lives. There is no informed consent per se. I mean, I think that you consent when you sign up for the registry as a participant, but there's not a provider that is administering the consent, because it's, you just sign yourself up, you self-enroll. But I can get you the specifics on the IRB.
Thanks, Dr. Gwathmey. So for the demographics, just wondering for your manuscript, are you going to report race and language that's primary? Was that gathered? And then also diving in more, generalized versus ocular is challenging, but more into, thymoma versus no thymoma, really trying to understand these subgroups.
Yes, we have all of that data. So we definitely have the race and ethnicity data that will be presented in the manuscript. That's in our tables. And then in terms of the thymus gland status, thymoma, thymic hyperplasia, and thymectomy, we have all of that prepared as well. It's tricky because when you ask a patient, do you have thymic hyperplasia? Many people don't know what that means. And so the questions were structured in a way that's like, did you have a CAT scan? Was it abnormal or normal? Were you told it was a thymoma or something else? So it is a little bit difficult to drill down on that, but we do have that data. Thanks for asking.
It was my question about thymoma or not thymoma. At least that they should potentially know.
Oh, I'm sorry. Hi. I thought I was waving over there.
Fine.
So,
yeah. It's just repeating. My questions were about thymoma or no thymoma. At least that if they had an operation, they will know. And then if that was a thymoma or not is another.
Yes, it's a very elaborate line of questioning to get to all of that specific detail about the thymus gland status, including the surgery, what the pathology was, and so on. So that's built into the registry questionnaires.
Very nice talk. I was really surprised about the high rate of unemployment that was detected. I mean, it's over 50% in this patient population. Do you know what the primary cause is? Is it related specifically to disability caused by the myasthenia, or do you think the mental health aspect goes into that as well?
That would be purely speculation. I think at this point, I can't necessarily state whether or not it was, mental health concerns related to unemployment. It really was just a question about unemployment, part-time or full-time employment status. And so you're right, we need to do more work in that area, but I don't have those answers today.
So you had a high incidence of depression and anxiety. Do you know how that compares with the general population, especially given that your MG-ADL is a purely subjective report of phenotype severity?
I don't. Does anyone in here know the current rates of depression in all comers in the U.S.? I just don't know the prevalence rates off the top of my head for 50%. So, we might be on track then.
Unfortunately, we are out of time for questions, so thank you, Dr. Gwathmey.
I'll be back.
For your talk. Lunch is now served in the Winter Palace Hall, so enjoy your lunch, and we will see you at 1 hour for session 3, platform presentations. Thank you.
And the executive committee is now meeting too in a room. Follow me. I know I can't explain where it is.
Our first presenter is Professor Geeta Ramdahari from the University College London, who will be talking to us about co-designing a strategy to engage people with neuromuscular diseases from racially minoritized backgrounds in research. Thank you.
Thank you very much. So it's great to present this on behalf of my little group who worked on this. So Rizik is a fellow, a neurology fellow with an interest in equality and diversity. And Babakir is actually a patient partner who was a co-applicant in this and actually guided us on the development of this work. So, those are my disclosures. So, in the UK, a big funder of clinical research is the National Institute for Health Research, and for a long time, they have very much encouraged patient involvement at all stages of the research pathway, starting from, when we're just setting priorities, through to study design, and then to dissemination all the way through. And more recently, I was invited to a launch event where they have now this Include project, which is about how do we engage people from communities that are underrepresented, and that covers lots of domains, mental health problems, rare diseases, which is interesting to us, but also people from with racially minoritized backgrounds and ethnic backgrounds. And so there's certain questions that they encourage us to answer whenever we're applying for one of their grants. So these are the questions here, and I became very interested in this one, number 4, because I had a few grants on the go with them. So actually, how do our recruitment and retention strategies and methods, how do they help people to engage? And so it got me reflecting on some of the work that we've already done with my research group. So I'm the principal investigator for the neuromuscular rehab research group. So I look back at some of our cohorts and just to see, well, what are we looking like? And what I found is even in our cohorts, even though I've got a bit of awareness of this, we still had very poor diversity. And of course, we have that intersection of people from minoritized backgrounds and also rare conditions in the cohorts that we're looking at. So people are dispersed around the UK. Oh, sorry, I think there's things popping up. People are dispersed around the UK, and so harder to engage with community groups, initiatives that you sometimes see with some of the more common diseases like diabetes or asthma, for example. And I remember Mike Hanna presenting last year that actually, when we look at genomic data, 86% of it globally is actually from European ancestry. And so he leads the ICGNMD, which is looking to address some of this through centres across the world and actually look at global equity for this. I also do work with the INC, and we've been partnering with Nicole Cresson leads the diversity committee, and there we looked at, and there's about 8,000 people who are part of the Inherited Neuropathy Consortium, so we looked at some of the data there. What does that look like? Again, we're looking at nearly 90% of people identifying as white, and actually we had very poor information on the groups that weren't, so we're actually doing a project now to see if we can improve how people classify. So just coming back to locally, to those points that I raised a moment ago, I do a lot of co-production work with our research group, so we co-produce intervention strategies. So we're very used to doing that, and I realised that actually it's no good me saying this is what we should do to engage people. Why don't I go and ask the people themselves, and they can tell me what will work for them. And they're definitely the best people to guide us. So the aims of this, we're a very small funded piece of research. It was funded by our Biomedical Research Centre at UCL and UCLH. So the first aim was to explore the experiences. You know, what are the challenges? What are the barriers? Why aren't people engaging? And then actually share some ideas with this group to then co-design. So let's come up together with some strategies to address this. So I did this through three workshops. The first one was just let's throw around some ideas. What are the challenges? What are the concerns? And then we then brought some of those ideas together to start to build the strategy. And then we debated, we agreed on some consensus on what it should look like. And we also planned other things like, are there training needs for people? And actually, how should we launch this? So with that first workshop, with focus group work, you don't want too many people, so we had 6 people, but I intentionally got people from lots of different backgrounds, and this is the rundown here. And so I presented this as a poster previous NMSG, so I won't go into detail, but if anybody wants to know what they're all in those speech bubbles, I can tell you. But just to home in briefly, so we wanted to look at what factors affected engagement. So there were things around knowledge, so contact with specialist clinics often helped, access to information could be a problem, understanding why they were being enrolled in research, and people gathering their own information. Shame was an unexpected theme that came up, people's personal shame of having a condition, and the response of family members, and that could sometimes be a barrier to people wanting to really stick their neck out and get involved. Communication could be seen as sometimes being problematic. People didn't feel that research would necessarily reach out to them. Accessible language and format, and this is where translation and interpretation comes in. But remembering that actually, you know, it's a personal choice. Sometimes people find that sometimes they just got enough to do to get living with their condition. And then trust came in. So either that their clinical care wouldn't be affected, but also there was a mix actually of trust and distrust in medicine, which actually also affected engagement. So we brought together the strategy ideas and we went through the consensus. And so I'll just present, and sorry, it's a little bit death by PowerPoint, but just to show you some of the suggested strategies that they came up with. And they felt that access to specialist clinics was a really important first point, but recognized that it could be challenging. So actually, are there virtual appointments and telemedicine that could help with that when people are dispersed around the country? When it came to actually individual studies, they advised that actually we do set up patient advisory groups, or PPI groups as we tend to call them in the UK. And actually with those groups who help us to conduct our research, get people from diverse backgrounds involved in that, not just their ethnic background, but also with educational level. And actually giving people lots of information about what's required for them to be involved, training them if they need to, and also cover their costs as well, out of respect for the time that they're devoting to help us conduct the research. And they were saying in terms of access, actually, how do we use some of the open resources that are available now that are sort of friendly, as they said, that health professionals and patients could access. So they did say GP surgeries, which is the family doctor. That can be challenging, of course, with rare diseases. But I think that one thing that I certainly thought was a great point is how we use our charities. We have the Neurological Alliance, the Rare Disease Alliance in the UK. You're looking at disease-specific online communities. How can we disseminate information on trials there? And actually having representation in our study materials, whether it's videos, leaflets, and then maybe having websites or YouTube channels for any big studies that are happening, that the information should be accessible. And actually, although we're all very interested in digital communication, but not forgetting paper-based resources, because digital literacy can be very variable. Definitely in IRB documentation, simpler language. Of course, we can be a bit constrained with that, but maybe having headline summaries, translation. But a really important one was actually how we use interpreter services in the consent process, just so there can be a check of understanding and people can ask. That they were recommended a degree of cultural sensitivity and diversity within the research team, certainly when a diagnosis being discussed as an entry point to research, making sure that people are comfortable, particularly because of these issues I said, like shame, that could be present. But actually, every individual will have their own experience, and we shouldn't make judgments about particular communities. They were very clear about that. And actually how we educate our research teams in this cultural sensitivity, and actually maybe even look for diversity within the team, particularly if there's a specific community we're trying to target. But giving incentives on the transparency, the purpose, that they said that wasn't always apparent to them. Expenses for travel, and also vouchers and payment, because some people maybe have a significant loss of income if they're coming to our centres and travelling. So actually, how do we reimburse that? But they felt that the contribution to society was also very important to highlight, and actually we're welcoming and we're pleasant, so, and the environment's nice. Almost there. So, and this one again was a surprise to me, but about how we involve family members in research, because in some communities the elders will actually help them make the decision or make the decision for them. So actually materials for them can be very helpful as well. And again, because of that, a range of formats, particularly if it's older people or people with less digital literacy. And then reassuring family about what is involved with involvements, reimbursement, etc. And then finally, how we communicate research outcomes. And people get quite annoyed. We promise it, and then it doesn't seem to happen. So actually prioritizing that in our study plans. And maybe how we use charities to communicate that using nice methods like webinars or online videos, and how we might actually use media professionals, or even we have social media influencers now who live with neuromuscular diseases. You see them all over Instagram now, and actually, how do they do it? How do they reach out? So, the group that was a very London-centric group, I think that was a limitation, and it may well be different in different countries, different geographical locations. And actually, this group were all very highly educated. They're all at least degree level. So I think that was a drawback as well. And not all communities were represented. That can be difficult. So actually, a bigger piece of work could be a series of engagement events. But certainly, what I would recommend if people do want to implement this is actually ensure there's educational as well as demographic diversity in the advisory groups, and actually review the strategy with those groups just to tailor it to the study. So if anybody's interested and I can share the strategy with them, that's my email. Thank you.
Thank you very much. We'll take questions. Yes?
Hi, thank you for a great talk. What do you think about the idea that patients are more adherent, do better with research participation, or more trusting if they're working with physicians or caregivers who share the same background? And does that play into, I saw you said one, encouraging members from the same ethnic background or whatever it is in your research team. Does that translate to the higher up view there?
I think there was a general opinion that representation, and I talked about the cultural sensitivity and representation in the research team helps, but of course, somewhere like London, it's very difficult to have every single community represented. But I think sometimes, if there is a concern, they might just feel a little bit more comfortable talking to somebody who maybe is from even just a similar background. I've certainly experienced that before. People have said, "Can I talk to you about something?" They're not necessarily from the same racial background as me, but they see that I'm from an ethnic minority as well. So that's anecdotal, but certainly that's something that did come through, that they certainly felt that it could be helpful. And if we can't do that, then certainly making sure we train people in that similar sensitivity and how to sort of check in with people.
Thank you for an excellent talk. I'd like to get your perspective on two aspects. One is the culture of the employer and the educational institution about research. Here in the States, employers will not allow time off. You have X amount of time for vacation, health, etc. And if you exceed that, you have a risk of losing your job. Schools do not allow students to participate. They do not make accommodations for that individual who would like to participate and has to miss class.
Yeah.
Is that attitude the same over on your side of the pond?
Perhaps less so, but I think it does happen. And I think that discussion came up when people were talking about loss of income, because sometimes people they might have to take holiday leave or unpaid leave to come and participate. So actually, I think that's why if we are willing to perhaps even with a voucher do some type of reimbursement, at least it's respecting the fact that they have to do that. I mainly work in adults, so there might be others who have more of the experience with children missing school, but certainly that discussion did come up, because not every employer is supportive.
Yep.
Thanks.
Dr. Harris?
So I have a question. I saw a really interesting talk about the ALS population and this similar types of ideas, and the person that was presenting identified geographic differences as a major barrier to participation and also possible contributor to differences in different pathologies, and I'm wondering if you saw the same.
I think that's why we need to do more. We were really London-centric, as I say, so I think we didn't have the spread of that. There's an application going in at the moment for a sort of a UK integrated network for a particular condition, and one of the things we're going to try and do is interview and collect information from people so we can see if some of the barriers are similar from all different parts of the UK and Scotland. So I suspect it probably is a factor, but this is where I think our national networking and collaboration is important in terms of people travelling. But yes, there may well be cultural differences depending on where you are in the country. I don't know what they are yet, but we might have more insight.
One more question. Was there anything surprising to you in the qualitative piece that you'd like to share?
I think it was the shame. And I'm from a South Asian background, and there's family members, and when I stopped and took off my researcher hat, and I then thought about my own community, I really recognised it, and that's where I think we sometimes have to stop being, Geeta Ramdari, the researcher, and actually remember what our own communities do and feel. Because I think we're a little bit in danger of that. We were from a sort of a paternalistic culture professionally often, and so you tend to sort of put that to one side. So it was a wake-up call, actually. It wasn't necessarily a surprise. It was a wake-up call to go, don't forget that actually this can be really, really significant for some people. The trust, I kind of was expecting that, because of vaccine hesitancy, etc. But the shame side of it was a good reminder, actually.
And that's something we can address even in the clinical encounter as well.
Exactly, yeah.
Yep. Other questions? All right. Well, thank you very much. All right, for our next talk, we're welcoming back up Dr. Kelly Gwathmey from Virginia Commonwealth, and she's going to be discussing a study of the common factors that influence fatigue in myasthenia gravis.
Thank you so much again for the invitation to present our work titled A Study of the Common Factors That Influence Fatigue and Myasthenia Gravis. I feel like I'm about to open up a can of worms here. I'd like to thank my collaborators from VCU and my neuromuscular division, as well as a number of different trainees who've helped me with this project over the last few years. Here are my disclosures again. So, introduction. So fatigue, as everyone in here knows, is a very common complaint in myasthenia, but poses many challenges in terms of diagnosis and treatment. So myasthenia is characterized by clinical evidence of fluctuating weakness that worsens with exercise and repeated movement, so muscle fatigability. But fatigue can be differentiated as either peripheral muscle fatigability or central fatigue, which is characterized by experienced lack of energy that limits mental and physical activities. So what do we already know about this topic? So fatigue in myasthenia is highly prevalent, up to 82% in some studies, and may not always track with disease control. And I want to pause here and say this is really why I got interested in this, because I became very frustrated when I was seeing patients in clinic with an MG-ADL of 0 and they were in pharmacological remission or had minimal manifestations, and they were complaining of this debilitating fatigue, and I didn't know what to do with them. And they were asking me to escalate their myasthenia treatments, and I just couldn't wrap my head around it. And so that really started me off on this journey to try and understand it better. Ooh, that went too fast. So in myasthenia, fatigue appears to be influenced by depression. We know that. Sleepiness and disease severity based off a number of different studies that are referenced at the end of this talk, and I'm happy to share the references if anyone's interested. We know that the worse the fatigue in myasthenia, the greater impact on quality of life. And we know in three recent phase 3 clinical trials, treatment of the myasthenia results in improved fatigue outcomes. But fatigue is multifactorial, as everyone knows, and it results in this vicious cycle of reducing physical activity, and then that in turn results in worsening weakness, and then that in turn makes the fatigue worse. And so my study here is to look at what comorbidities also could be contributing to fatigue that potentially we can target and make people better. So the objectives are to study fatigue in seropositive and seronegative myasthenics and correlate this with disease severity, which has been done in other studies, and to explore other factors associated with fatigue in MG patients, and key, identify treatable drivers of fatigue to guide MG providers, such as you in the audience, in the clinical management of these patients. So methodology, we basically approached anyone in our clinic that complained of fatigue and had myasthenia and asked if they would like to join us and enroll in this study after signing a consent, and it was through the IRB. So we had 46 MG patients, both ocular and generalized, seropositive and seronegative, with fatigue. So I did not use a fatigue scale to screen them to get into the study because I really wanted this to mirror what we're all encountering in clinic. And so there were a number of different things we collected on these patients. We collected labs, and the rationale behind the ones that I chose were a CBC to look for anemia that can cause fatigue, B12 and methylmalonic acid, 25-hydroxy vitamin D levels, TSH, and testosterone in the men. We administered the MG-ADL, QOL-15R, the composite. We had the fatigue severity scale and the neuro quality of life fatigue scale as the two fatigue instruments. And then for anxiety, the neuro quality of life anxiety scale and the neuro quality of life depression scale. And they're the full-length versions of these scales, not the short forms. And then the Pittsburgh Sleep Quality Index. We did also use the Epworth Sleepiness Scale, but it did not perform very well, and I won't be presenting that data here. So in terms of the demographics of our 46 patients, the average age was 59, the disease duration was just over 9 years, and almost 74% were women. So 80% were AChR positive, And so what we did was we asked the patients, what were your symptoms at time of symptom onset, and then what were your current symptoms at time of enrollment into the study? And it was a single visit. And so the symptom distribution onset generalized in 82.6% and ocular in 15.2%. And then the current symptom distribution, what I'll point out to you here is that we have a total of 38 out of 46 patients that were reporting current symptoms. That meant the rest of the patients were asymptomatic. And so almost 70% had sort of generalized symptoms at time of enrollment and 13% had ocular. The average MG-ADL score was 4.7, and the average MGQOL-15R score was 10, and the composite 5.84. So first we start off with correlation studies using Spearman correlation analysis to look between these different variables in this fatigued MG population. And what did we find? First of all, older patients have less anxiety, less depression, and less fatigue. And you see the p-values in red there. So the fact that older patients tend to have less fatigue has been reported in other studies. We did find that lower vitamin D levels were associated with greater fatigue and depression. And I'll just stop here and point out that of our 46 patients, 15 of them had vitamin D deficiency by our lab criteria. So that's all a third. So that was quite impressive. So also in terms of lower vitamin B12 levels was also associated with fatigue, and we'll get to this more in a moment. And then the PSQI looking at sleepiness was really associated with greater anxiety, if you see the p-value at the bottom. So this is busy, and bear with me. So what we then did was we used multiple linear regression modeling to put some other variables into the analysis and look at the performance of like vitamin B12 deficiency and vitamin D deficiency in relation to fatigue here in this model. And so again, we used the Neuro-Quality of Life Fatigue Scale and the Fatigue Severity Scale. And so what we found when we were adjusting for age, disease duration, sex, disease severity, and sleep quality in this model. And so we did find, similar to other studies, that the MG-ADL or disease severity did correlate with worse fatigue on both scales. So for every point increase in the MG-ADL, you had a 2.2 point increase on the Neuro-Qual Fatigue Score and a 1.4 point increase on the Fatigue Severity Scale. And then also in this model, we found that sleepiness correlated with the Neuro-Quality of Life Fatigue Scale as well. So then in the bolded underlined part for B12 deficiency, so we considered someone B12 deficient if their B12 level was below our lower limit of normal or their methylmalonic acid was above the upper limit of normal for our lab. And so you were yes if either of those was true. And so if you had B12 deficiency, your score on the Neuro-Quality of Life Fatigue was 6 points higher, and then it was 14.4 points higher on the Fatigue Severity Scale. Now here you'll notice that when we considered all these other variables in the model, the vitamin D deficiency did not clearly correlate with fatigue. All right, and here's the last model I'm going to show you. This is looking at the association of depression and anxiety with fatigue, and this also adjusted for the same variables. And so first I'll point out that men are just less fatigued than women. So with the fatigue severity scale, you'll see that it was just much less common to have fatigue if you were a man. And that's been reported as well in other studies. And so same thing here with the MG-ADL looking at disease severity with this model taking into consideration depression and anxiety, there was also still association with fatigue. Now the interesting thing in this model, the association between sleepiness and fatigue goes away when you put depression and anxiety in the model. And then we do see, again, association with depression and fatigue with the Neuro-Quality of Life Fatigue Score. So our conclusions were that fatigue is more commonly encountered in myasthenia patients who are younger, who are female, and have more severe disease activity. Fatigue is associated with depression, less so anxiety, and fatigue is still present despite accounting for disease severity and mental health concerns in our modeling, suggesting that there are other factors. So upon testing for these additional causes of fatigue, B12 deficiency was highly associated with fatigue. Vitamin D deficiency clearly associated with depression and fatigue, but once you account for the other variables, vitamin D no longer was associated with fatigue. So we think perhaps the depression's driving that. And then sleepiness did not correlate with fatigue when depression was also considered in the model. So the recommendations from this: in fatigued MG patients, screen for depression, screen for vitamin B12 and vitamin D deficiency. I mean, why not? This is low-hanging fruit. We all can do this in clinic. These are treatable causes of fatigue. Fatigue is tightly linked with disease severity, and recent phase 3 trials suggest improved disease control results in improved fatigue. And so next steps. So larger prospective studies certainly are needed to look at which factors are causing fatigue, not just what's associated with fatigue. And then we also did, and I didn't show all of this data, we explored the association of medications such as corticosteroids with fatigue, but our numbers were simply too small. Thankfully at VCU, we don't have a lot of patients on high doses of corticosteroids, so we couldn't really comment. But also, we looked at like MGQOL 15R, as I mentioned, and I didn't present that data here, and we still need to do further analysis on that. So with that, I'd like to thank you for your attention. I'm happy to take any questions.
Thank you. Dr. Amato?
Did you look at sleep apnea?
So, I mean, we did just the upwards sleepiness scale and the PSQIs, the two scales. Now, I do have all the medical comorbidities separately for every patient that was enrolled, but we have not looked at that and pulled it out yet. Good question.
Also, a diagnosed sleep disorder in general, if they were on stimulants, for that or otherwise.
Right, excellent.
In the back?
Or
it's kind of hard to see. I can't see where the, okay, over there.
Yes.
Great.
Hi.
Hi. Thanks. That was a great talk. Great work. I'm over here.
Sorry. Oh, gosh. I can't see anything. Thanks.
No, really nice work. I'm not familiar with the myasthenia gravis composite score or the ADL score, but so forgive me if for the question if I'm wrong. But does that include an assessment of muscle fatigability? And if not, how do you think understanding muscle fatigability and its relationship to fatigue, or how it might have influenced your results?
Right, that's a great question. No, we did not include any instruments that would test muscle fatigability specifically. And so the composite's like a weighted scale that includes the physical exam and some patient-reported symptoms.
Is there a relationship in the literature of fatigability and fatigue in it?
I think there is a study that looks specifically at muscle fatigability objectively with fatigue, but I would have to go back and pull that. Sorry.
Great, thanks.
People may have done that study.
Right there.
If I may.
Hi.
Oh, yep.
In addition to the sleep apnea, I was going to ask if you recorded, I'm sure you recorded the weight or BMI.
Yes, we have all of that data. Yes. We haven't analyzed it
and
yet.
I was just going to comment on the age. So when you say older people seem to have less fatigue, females more fatigue than men, I wondered whether lifestyle and being older and they accepting more the disability that they have, not being so bothered about the fatigue than younger people, very active, multitasking, etc. Whether younger women suffer with more fatigue also because of that, not only because myasthenia, but could be factors that could contribute to that.
Right, I absolutely agree with you. I think it's going to be very hard to unwind this, all the different variables and contributing factures.
Maybe looking at age categorically to see if there is a change with older age.
And I agree, and I think we need more numbers. I mean, 46 people is not, I think, enough to get a really good sense, but okay, sorry, I think there's another.
Thank you so much for sharing the study. Quick question, going back to your original statement from your study, it might be too early to answer, but like the statement you made, like patients who have minimal manifestation or pharmacological remission, how do you put your study findings to explain that fatigue in the patient population of MG?
Well, I mean, I will say it was very eye-opening coming out of this, how many patients we identified that had depression when we screened for it, and patients who had B12. I put everyone on vitamin D after this, I've got to tell you. And so it just really opened up my eyes. And so that was my takeaway, is I need to do a better job of looking for easy, identifiable causes of fatigue, especially in patients who are otherwise doing well.
Great.
You're next, and then-
Could we raise the lights just for the Q&A just a little bit so that we can see? Okay, right there.
Over here next, and then these will be the final two questions.
Great. Okay.
Great work. So my question is, I'm not familiar with an instrument that's used to screen for depression. Does it have both somatic and affective symptoms as a screening for depression?
In the questions, you mean?
Correct.
Yes.
Because that sometimes might falsely inflate the rate of depression because you're basically including the somatic symptoms.
Fair enough. Thank you. Yeah, and we tried to use instruments that were like free and in the public domain, just because this was unfunded. Yeah.
All right, one more question.
Final question.
Thank you. I'm interested, it comes back to the question about age actually, and also what Jackie was saying about, fatigue is multifactorial. You have peripheral fatigue and fatigability, but you can also have central fatigue, which has been looked at in other neuromuscular conditions, and we know that central fatigue is lower in older people as well, so that's another factor. So it would, I'm just wondering, in terms of the next step from this, would there be an opportunity to explore the nature of fatigue in more detail, like what's been done in other, because I know the group in the Netherlands have done a lot of work in that.
Right, you're exactly right. So yes, I mean, my next step would definitely be to secure some funding to support this work, because honestly, just to take a patient through this process was about 2 hours of questionnaires and labs and what have you, and they were doing that just of their own goodwill. And so I think in order to do more, the fatigue impact scale, I believe, is a 40-item instrument. It breaks it down between, right, I think cognitive fatigue and physical fatigue. And so using other scales would be helpful too, to sort of drill down. And maybe, as mentioned, objective tests of muscle fatigability would be extremely helpful. Yeah. So thank you for your great questions.
Great. Thank you, Dr. Gwathmey. All right, for our next talk, we're going to have Professor Eric Voorn from the University of Oxford, and he will be speaking on combined, personalized, home-based aerobic exercise and coaching to improve physical fitness in neuromuscular diseases, a multi-center, single-blind, randomized control trial. Thank you.
Thank you for the introduction. I'd really like to go to Oxford once, but I'm actually from Amsterdam UMC, but it's wrong in the agenda. I know. Having said that, with all sitting us here all day, it's great to be given the opportunity to do a talk about exercise. So I'm really excited to share with you some results of a randomized controlled trial that we just finished. But before I start, I would like to acknowledge that the name of Sander Oorschot, who is actually the PhD candidate that I supervise, and who actually did all of the hard work. We did this trial in times of COVID-19, and you may have experienced as well that the government sometimes took measures, for instance, closing of local gyms, which really made this an extra challenge to complete it. So I'm really proud of him. Having said that, so these are some disclosures. Well, coming back to the previous talk, there was also a vicious circle mentioned, so it's good to see the complaint of fatigue here as well. So the idea we have behind prescribing exercise to people with neuromuscular diseases is that these symptoms, such as fatigue, but also muscle weakness and pain, that they actually contribute to physical inactivity, and we know that that leads to deconditioning, and that again may worsen the symptoms, etc., etc. So the idea of using exercise as an intervention is to actually break this vicious circle of inactivity by trying to improve the reduced physical fitness. And in recent decades, there have been numerous studies showing the beneficial effects of aerobic exercise for various neuromuscular diseases. But, there is a but, there have been several systematic reviews recently And they all identified that there is still a need for more high quality evidence. And that has to do with the fact that most of the studies that were done, most of them were actually uncontrolled, underpowered, or lacking intention to treat analysis. So there is really still a need for more high quality evidence. And the other important finding was that there wasn't really attention for the longer term effects. And that was not only the assessments on the long term, but also the focus of the intervention, because most of the studies, they just focused on exercise itself, but not having attention for that. I mean, in the end, what we want to achieve is that people remain having a physically active lifestyle. So there should also be a focus on that as well. So with that, we designed an intervention that focused both on aerobic exercise to improve physical fitness, but it also included a coaching program that was really aimed at sustaining an active lifestyle on the longer term. So the primary focus of this study was on physical fitness, and then we also had some secondary outcomes that are on the screen that you can read yourself. So this was the design. So it was a multi-center trial, so we had 6 different centers in the Netherlands. It was a single-blind study, so it's difficult to blind participants, but the assessors were actually blinded. And people were randomized to an intervention study or a control group. In the control group, they received usual care. And in the randomization, we stratified for center as well as for diagnosed groups. I will come back to that later. So we included participants with neuromuscular diseases without any effective drug therapy, and being expert center for post-polio syndrome, which is a motor neuron disease, CMT, which you all know, and also inflammatory myopathies and congenital myopathies. These were the main groups that we included. So we searched for people who were motivated to improve their reduced physical fitness, and we did this study in adults. And as you can see, we did assessments not only directly post-intervention, but also 6 months after the intervention and even 6 months later, so up to 1 year after the intervention. So what about the intervention? So it had 2 different components. I already mentioned that. It had an aerobic exercise program, was mainly home-based, and it was personalized in the sense that we used the results of exercise testing to prescribe exercise. And being an exercise physiologist, this is something that I can give to you, is when you usually prescribe exercise, these are the factors that you need to address. They are called the fit factors. So first think about the frequency, so in this case 3 days a week. Then you have to say something about intensity, and when we talk about polarized exercise, that means that the majority of your training volume is actually at the lower exercise intensity. So you have to think of about 75% of the total training volume was at low intensities. And only 25% was actually at higher intensities. And what made this different from most of the previous studies was they are often called moderate intensity aerobic exercise. And also what they do is they prescribe exercise at a fixed intensity for the entire group. So for instance, at 60% of your maximum heart rate. And what we did differently in this case is, I mentioned that we used the exercise testing results. More specifically, we looked at the first ventilatory threshold, which is a marker that really differentiates between lower and higher exercise intensity levels. And we know from previous work that there is a large variation in this group if you talk about this this measure. So this is really a very personalized approach. So in terms of time, you see 20 to 40 minutes. You can maybe imagine that if you do high intensity training, that of course it's also shorter because you can just do it shorter. And it's also longer if you have lower intensity sessions. And the type of exercise in this case was on an ergometer. And the reason for that is that that really allows to manage the workload very well, which is important for this population. So it was a home-based program. We also developed an app specifically for this program. And people were provided with a heart rate monitor that could actually connect with the app. And people really got real-time feedback on their target heart rate ranges. So that made it more easy for them to actually keep training within these limits. And the other positive thing is that for the supervising physiotherapists, it was also very easy because immediately after an exercise session was finished, they could have a look at the training results, which is very useful if you monitor training and have to give feedback. Earlier we did some pilot working both on the exercise program as well as on the usability of the application, and that was already published, as you can see. Then in short about the motivational interviewing coaching program. So this was done by occupational therapists, and so as you can see, there were some face-to-face sessions, but also some telephone sessions. And motivational interviewing really means that you're not going to tell your patient that it's good to exercise and to have an active lifestyle. But you really try to let your patient think, okay, what could it potentially bring me if I have an active lifestyle? So they really have to come up with their own goals, their own plan, and these are just tools to help them. So about the outcomes, so the primary outcome in this study was the peak oxygen uptake. It has been used quite often in exercise studies. It's what is called the golden standard for measuring your aerobic capacity, which is one of the physical fitness markers. I can also imagine that not all of you know what it is. So what you do basically is you put someone, in this case on a cycle, in the Netherlands we like cycling, so we put someone on a cycle, and then you just, you measure the gas exchange variables, and meanwhile you just increase the workload until the patient is no longer to keep up with that pace, and from that you can actually determine your maximal oxygen uptake. And we also have done some work on that as well to show the validity of this outcome in neuromuscular diseases. Basically, our primary outcome was directly post-intervention, but of course we also looked at the longer-term effects. Secondary outcomes, as you can see, included daily activity, quality of life, physical functioning. Also, we looked at metabolic risk factors. So one of our ideas was, okay, if we have a longer-term follow-up, maybe that also has consequences for metabolic health. So we also took cholesterol and glucose, but also blood pressure, for instance. Creatine kinase was done to track muscle damage, and of course adverse events. And we used an intention to treat analysis based on linear mixed models. So what about the baseline characteristics? So I mentioned that we were an expert center for some specific neuromuscular diseases. So you can see that we came up with 3 different groups. The distribution across the control intervention group is quite equal. So randomization worked well. To give you an idea about these values of peak oxygen uptake that you see, so let's say around 20 milliliters per minute per kilogram. You have to imagine that if you have a reference group, so a healthy group for this particular age, they would be above 30. And also in literature, if you are below 20 milliliters per minute per kilogram, that is believed to lead to independent functioning in elderly. So that's just to give you an idea of these numbers. Then the most important, of course, so what about the intervention effects? It was a positive trial, as you can see. So we found an increase in VO2 peak directly post-intervention, and it was a 10% increase, so it was substantial. And also what you can see was that there was an overall average time effect. At the same time, you also see that the effect seems to fade away a bit over time. I also want you not to focus too much on the p-values. Of course, they are important, but what I want you to really look at is really the intervention effects, the effect sizes. So you can see that they just seem to fade away a bit. Why I don't want you to look too much on the p-values is that they are also influenced by the number of people, and you can see that there was, we lost some people along the way, and it also had to do with these COVID measures because we simply couldn't do the measurements in some cases. So for the secondary outcomes, we found no significant differences between groups for any of the secondary outcomes, and that included adverse events and also creatine kinase levels. So in conclusion, we can say that this combined program was safe, and it also improved the physical fitness in a group that was deconditioned, but we did not find conclusive evidence for improved daily activity, quality of life, and physical functioning. So I think the home-based approach really makes it a good target for wider implementation. It really has potential, and here are also some ideas for future research. There's two closing remarks that I want to make. So if you are interested in our work, you can access the website that we have. We have written all the training protocols in what we call the BFit training guide. It is freely accessible. It is really some practical tools for healthcare professionals, for physiotherapists on how to prescribe exercise. There's also some background information about why to prescribe exercise, but really how to do it. And then last but not least, this is also something that I'm really enthusiastic about. First weekend of October, I'm going to organize a NMSG workshop on exercise. One of the co-organizers is also here, John Fissing. Well, one of the things we're currently doing is we're doing a survey, both among patients. And patients, we already have over 2,000 respondents. But we're also doing a survey among healthcare professionals. I assume that there are many of you here, so if you're willing to do it, please help us and fill in this questionnaire. Even if you do not prescribe exercise, but you do have ideas why you don't do it, there is room in this questionnaire to inform us about that. So having said that, I'm going to close this talk, and I'm happy to take any questions.
Great, thank you. We have time for one question, I think.
Oh, this is great to see, Eric. And it's interesting because actually the percentage change that you have, because when we did our aerobic study in CMT and IBM, it was between 11, because we looked at the two groups separately, it was between 11 and 14, so it's similar magnitude. But you know when you were saying that the T2 and T3 results faded, was that compared to baseline or was that compared to T1? Because is it that people just plateaued or was it people continuing? What do you think that was?
So visually what it does is there is an increase from baseline to directly post-intervention, and then it fades away a bit. So it's more like this, yeah. Slightly fades away. Yeah.
Great, thank you very much. All right, and for our last talk of the session, we're going to welcome Professor Pietro Zara, and he is from Oxford, not the University of Amsterdam. And he's going to present on an analysis of mortality rates and causes of death in an Oxford cohort of adult myasthenia gravis patients. Thank you.
Good afternoon, everyone, and thanks for the invitation. It's a pleasure to be here and to present our first study about mortality rates and the causes of death in an Oxford cohort of adult patients with myasthenia gravis. As you know, these are my disclosures. As you know, myasthenia gravis is a rare autoimmune disorder characterized by antibody-mediated dysfunction of the neuromuscular junction, leading to abnormal muscle fatigability and weakness. Approximately 85% of patients with myasthenia exhibit antibodies against acetylcholine receptor, while a minority of them display antibodies against muscle-specific kinase and other less common targets. Clinical manifestations vary among patients with myasthenia, with 15 of the patients having poorly ocular symptoms, while 85% having more generalized muscle weakness. Furthermore, 50% experience refractory disease, and approximately 15-20% experience myasthenic crisis in their lifetime. However, advances in medical care and therapy have significantly decreased the mortality rate due to myasthenic crisis, dropping from 80-50% in 1960 to 4-6% in recent years. And the current reported mortality rates of myasthenia range from 0.06 to 0.89 per million person year. The mortality data about mortality vary in literature and are controversial. There are some epidemiological studies suggesting that patients with myasthenia have a favorable prognosis and a long-term survival rate similar to the general population. In contrast, other studies suggest that patients have a higher mortality rate and reduced life expectancy compared to the general population. Causes of mortality reported in literature are very and scarce, making comparison between studies extremely challenging due to differences in population demographics, economic resources, and methodology. For example, in this Swedish study, the most common ultimate cause of death was any cancer, followed by ischemic heart disease, myasthenia gravis itself. And in contrast, in these more recent epidemiological studies conducted in China, the primary cause of death was myasthenia gravis itself in approximately 85% of patients, followed by cancer, other neurological disease, and ischemic heart disease. The aim of this study was to assess the risk of mortality in the Oxford cohort of myasthenia gravis patients and compare to the general UK population. This is the first study because we don't have other studies about mortality before this. And the other aim was to identify the leading causes of mortality to better inform future clinical management. In order to collect this data, a single-center retrospective mortality analysis was conducted in a cohort of 744 adult patients with myasthenia gravis who were actively followed up at Gerard Cliff Hospital over an 11-year study period. Mortality data were obtained from our database at the Gerard Cliff Hospital and supplemented by patient folder review and the GP records. And finally, a standardized mortality ratio was calculated for the Oxford myasthenia gravis cohort as the ratio between the number of observed deaths in the study population and the number of the expected deaths. So over an 11-year study period, 88 out of 744 patients deceased, with 60 of them being male. The mean age at death was 76.8 years, and the time from myasthenia gravis diagnosis to death was 13 years. In this table are summarized some clinical and demographic characteristics of our cohort of patients, with the majority of them having late onset myasthenia gravis. Roughly 25% had thymoma, and the majority of them had SHR antibody positivity and generalized myasthenia. Moreover, 65.5% of patients was treated with steroids. Azathioprine was the second most commonly used immunosuppressive agent, followed by mycophenolate, and notably 65.1% of patients did not receive any immunosuppressive agent. We found an overall standardized mortality ratio of our cohort of 1.20, suggesting an increased number of observed deaths than expected. And in the age subgroup of 40-49, we found a higher standardized mortality ratio. And interestingly, this group consisted solely of females. We identified about causes of mortality. The leading primary cause was malignancy, followed by cardiovascular diseases, infectious, and interestingly, not one patient over 11-year study period died due to myasthenic crisis. More in details about the frequency of different cancer types, we found that the leading neoplastic causes of death were gastrointestinal tumors, followed by thymoma and lung cancer. We identified also an early mortality subgroup defined as a subgroup of patients who died before the age of 65. And this group was made up predominantly by females. And we found an overall increase in all types of malignancies, thymoma, and lung cancer-related death. And we found also a higher associated history of thymoma. In conclusion, in the early mortality, we observed that they are mainly associated with thymoma, lung cancer, and female sex. Also, cardiovascular disease impact on mortality, and patients who died due to cardiovascular diseases were more commonly linked, associated with two or more of the following risk factors, such as hypertension, hypercholesterolemia, and diabetes. The majority of them were older males. As we know, immunosuppressant therapy is associated with an increased rate of mortality and morbidity. But surprisingly, we found in our study that steroid therapy was not associated with duration of disease, age at death, and increased rate of cardiovascular related death. But we found that azathioprine seems to be associated with an increased rate of mortality due to gastrointestinal tract malignancies, but not with overall malignancies. We also know that immunosuppressant therapy can increase the risk of infectious. In our cohort, the most common infectious were respiratory and urinary tract infectious. But interestingly, we found that only steroid therapy was associated with an increased risk of infectious, and not similar association was found for other immunosuppressive agents. Finally, we identified another subgroup of short disease duration, defined as 5 years or fewer between myasthenia gravis diagnosis and death. And it was associated with a higher mean age at diagnosis, with the male gender, and the majority of them had the late onset myasthenia gravis, 92.3% of the cases. Interestingly, not using steroid-sparing immunosuppressant drugs was associated with a shorter duration of disease. In conclusion, we can say that in the Oxford myasthenia gravis cohort, the overall mortality ratio slightly overcome that expected in the general UK population, that malignancy was the leading primary cause of death in our cohort, with many thymoma cases driving early mortality, especially in younger females. That the possible contribution of azathioprine treatment to malignancy risk in myasthenia gravis cohort should be further investigated. And finally, that high-risk patients with multiple comorbidities should be identified early and treated for a clinical suspicion for anticipated clinical complications. And thanks for the attention.
Thank you very much. We have a couple minutes for questions. While we're walking the microphone over, have you thought about doing the analysis separate between thymoma and non-thymomatous patients?
Not, but we are going to do it because we haven't finished the study yet. And yes, it's interesting. We found just that thymoma was associated with females, mainly with females and young females. Yeah.
Yeah, and I was going to ask, it just seemed like there's a very high rate of thymoma, and in the patients who died, were they exclusively malignant thymomas, or was it both?
I don't know, because I don't have now this information about the severity of thymoma. Are you? Yeah, the classification. No,
pathology.
We don't have the information about pathology, but it would be interesting. Yeah, it would be interesting to focus on this topic. Yeah.
Hi, I was just wondering. You mentioned about lung cancer being being highly associated. Did you differentiate like different types of lung cancer, since we know there's types that are more prevalent in people who don't smoke and don't have those risk factors?
No, we don't, but it's interesting also because we found association with the lung cancer, especially in the early mortality subgroup, so in patients younger than 65 years old, and this is not linked with what happened in the general population, in which the tumors, the lung cancers, are more frequent in the elderly patients. So it will be interesting to study this.
Do you know about the azathioprine, the duration of the treatment with the malignancy association you found? Just curious. The azathioprine you mentioned, because that is something interesting, like to know, like the duration of the treatment.
No, we don't have this information. Yeah, but it's interesting also because there are these mirror other studies, especially studies about bowel inflammation diseases, in which they found a similar association between azathioprine and gastrointestinal tract tumors. So it would be interesting to see how long the patients are treated with azathioprine before developing the tumor.
And presumably shorter than disease duration, at least. So you have that time point, right?
I was also wondering about dosages of the medication, or the dosages of the medications that were prescribed, and also if patients on combinations of immunotherapy may have had higher rates of malignancy.
The combination, in particular, the treatment combination is in particular with steroids and another immunosuppressive agent. But I think that especially steroid-sparing agents are associated with an increased risk of cancers or neoplasms.
Thank you. It's great work. So I'm not sure if I got that right, but did you mention 68% of the patients did not receive immunosuppressive medications?
Yeah.
So what's your explanation for that?
Yeah, it's a great number. Maybe it can reflect a group of patients who died before starting the stepwise treatment. Maybe the majority of them are patients with a shorter disease of duration. Yeah.
Question, great study. Do you have any comments you could make right now about the dates people died? You looked after a large cohort of immunosuppressed patients through COVID. Did more people die then than you would have expected them to be distributed through the rest of your study?
Only one patient died during the COVID period, so it does impact significantly as one of the main causes of death in our cohort.
Thank you.
Thank you for the presentation. I was wondering if you accounted for patients' habits, such as smoking and other potentially risk-associated activities, considering this unexpected high, at least in my opinion, frequency of...
No, we didn't consider it because it's a retrospective study and it's also difficult to collect this data about lifestyle, patients' lifestyle.
All right. Thank you so much.
Thank you very much.
All right, if everyone could please take a seat for the afternoon Young Investigators session. Really, I mean, it's an early career researchers session, so thank you for joining. So we've got a nice little afternoon lined up with a few talks to start about grants and accessing research. We've got a quick icebreaker session, and then we have some breakout groups after that. So I'll hand you over to Mike.
So it's our honor to have Dr. Lauren Rioma from the NIH joining us today to give the keynote address for this session. She is the director of the NINDS Clinical Trials Unit. She oversees internal clinical trials. She did her neurology training at the University of Florida and then did fellowships in neuroimmunology and clinical trials methodology at the NINDS. And I got to meet her when she was co-directing the experimental therapeutics section of the American Academy of Neurology, and she wrote a really awesome review. If anybody hasn't seen that in Continuum from 2 years ago now, you should check it out. It's a really good primer on doing clinical research, so it's an honor to have you come speak to the group today. Thanks for coming.
It's an absolute honor to be here today. This is my first time attending this meeting, and it's just been fabulous all day, so I really appreciate the invitation. A lot of fun. This is a topic that's a bit near and dear to my heart as a federal employee, talking about intramural NINDS and supporting experimental neurotherapeutics as a research mission. I'm perpetually, I add too many slides to my talk, so that's going to be true here. So we're going to zoom through a couple of different slides as we walk through a little bit of this setup of intramural NINDS. I said on the intramural side of NINDS, you might be more familiar with the extramural side of NINDS, so I'm going to walk you through that a little bit. I'm going to walk you through some core concepts that we looked at when we started building research support services for multiple different PI groups, and kind of our field of dreams, what we were envisioning that we needed within our program. I'll zoom over some slides with some central tenets of policies that have changed that are applicable to NINDS. Much of this will be applicable to NIH type research because it speaks to NIH policy. If you are implementing pharma-only, non-federally funded research, some of this policy might not apply to you. And at the end, I have a couple of highlights of lessons learned. I think we probably all have 10,000 lessons learned from clinical research. So I just highlighted some that either spoke to some literature concepts, other things that maybe you could take home, look up yourself, and see if they would be of benefit to you. And then I posted some kind of research training opportunities that I'm aware of. They're certainly not comprehensive. There are many good ones out there. These are just a snapshot. So that will be the talk that we walk through today. So the NIH Clinical Center sits in Bethesda, Maryland. It's huge. It sits right across from Walter Reed. It's larger, I think, than the Walter Reed campus. And the body of it in Building 10 is a purely clinical research hospital. So nobody is seen in this hospital that's not on a clinical research protocol. Every single person there is contributing to research, and they're participants on a protocol, even if they're also patients that are seen in the hospital. There's no ED. We get our patients from word of mouth, from collaborative studies, from investigators at other academic partnerships, pharma partnerships, other industry. 200 inpatient beds, 93 hospital, otherwise a typical hospital. We have a lot of shared resources that speak to research. So we have huge apheresis departments that can take and sort any cell that you're potentially interested in. Large investigational pharmacies that do very niche things, cellular, gene-targeted therapy, you name it, we probably have it in those realms. Huge radiology and NMR centers that span 0.06 Tesla all the way to 11.7. And I should say France got their 11.7 up before we did and have good data, so we're really looking forward to seeing all that French data come out. And this is the NMR center, so you can see quite a few magnets that are used for MEG, used for fMRI, tractography, and just used for standard imaging. At NINDS, in the intramural program, we routinely, on average, have about 126 active protocols a year. We are focused on early phase. We have in the entire program, on average, 1 phase 3 clinical trial. So we're heavily, very early phase, phase 2A is probably one of our latest phase trials. We have a variety of clinical, non-clinical faculty, same as any other academic institution, and a variety of trainees. And we have an office of the clinical director, which directs the clinical side portion of the research. There's also an office of the scientific director. And although we have had for many years PI groups or branches or sections that were discipline-specific, neuromuscular-specific, neuroimmunology-specific, there wasn't, around 2013, there wasn't any core resourcing that really supported work of clinical trials in a centralized fashion. So when our new clinical director came, this was a core mission that he put together, starting with an FDA NINDS fellowship. Oops, sorry. And then coalescing, you can see here, I put some pictures of our staff and how it's changed over time. And in 2014, about 3 years after our clinical director started, our newest clinical director, and a year after he created the FDA fellowship, they coalesced the staff that could be centralized within the institute. These are primarily research coordinators, protocol navigators, that's a support staff position that we have that really walks the protocol, the clinical research protocol, through the variety of regulatory bodies, IRB, IBC, radiation safety, et cetera, and put them in a unit together called the Clinical Trials Unit. Over time, we added to this unit. Until today, we have over 40 members and 7 units with dedicated biostatisticians, dedicated research coordinator teams, research coordinator training teams for other research coordinators within the institute, protocol navigators that are centralized, what we call patient care coordinators, or that person that really interfaces with the patient for their scheduling needs, pharmacovigilance arms, regulatory arms, and kind of the bread and butter of what you need to run a clinical research program. And this was an organic exercise. So every time a new policy or a new need came about in the institute, right, whether it was the revised comment, right, each of the changes in the final rule, new NIH policy that spoke to what was in line with what we used to call an applicable trial, right? But the NIH policy expands that. There are more things that they consider clinical trials. Needs for 21 CFR compliant databases when we took over sponsorship of trials. And so each of these needs led us to grow our core group of staff and to start structuring them in a way that made sense in a central fashion whereby they could support multiple other research teams. So today, I think I mentioned we have 7 units, and these are the core disciplines that we really target in a clinical research team. We want to be able to train our research support staff. We have a variety of trainees. They need trained. We need to be able to support them. We have clear job delineations and duties, have clear auditing plans, clear monitoring plans, and monitors and auditors that can do that. Integrated biostatistics at the very beginning of the protocol all the way to the end in CT.gov reporting. Regulatory and medical affairs, oversight for adverse events, oversight for DSMBs, centralization of DSMBs, compliance with policy, pharmacist that also sits with the protocol through the entire life cycle, an investigational pharmacist, not a clinical pharmacist, who sits with a protocol throughout its life cycle and helps source, helps design, the different encapsulations and a variety of therapeutics and checks the AEs, as well as administrative support for all this. And we support the NINDS research mission. We are all federal workers or contractors who support the mission of NINDS to help ameliorate neurologic disease. So clinical research is a team sport. Nobody can do this alone. No person is an island here. And when we think about a clinical research team, these are the core areas that we try to support. Every clinical research team is going to look a little bit different depending on the needs of the institute, the funding available, and the jobs at hand. It'd be very common to have a research coordinator do some protocol navigation and help route a protocol in a traditional small research team. And that was not unheard of with us either. But as we grew, we started to role delineate a little bit more and try to give people clear roles and responsibilities on the team. We also, because of the nature of NINDS, we have patient administration assistants, or we call them patient care coordinators, who directly interface on scheduling the patient. And the research communicator will tell that member of the staff the plan in accordance with the protocol and the protocol-specific procedures, and then the patient care coordinator will interface with the patient, help schedule them, help give them their reimbursements, etc. We've started to integrate, to the best of our ability, data managers into teams, and this speaks to some new changes with data management plans and sharing that have come out of NIH and the government in the last year or two, to try to have data as integrated as possible into the research plan, the team's research plan, the study team's research plan, for dissemination at the end of the study, and for a clear transit of where they're going to put data, where they're going to take it out, and how they're going to build it into their database. And then, of course, the research participant, because none of this could be done without participants in research. So these are some of the slides I'm going to skip over. I'm very happy to share any of them. And research needs a village, right? So all of those core components that we started to build into the CTU, those are essential to research programs. And whether they're independent people, or they're shared, or they're found through collaborations amongst investigators, they're all necessary to carry out different types of clinical research, depending on what type of clinical research you're performing, what level of trial, and how intense these things are. And oversight, regulatory oversight, protects you as an investigator and a coordinator. It protects your participants. It protects the ethics of the research. And hopefully it protects your data that you're going to give to the community to further research neurologic disease. So the IRB, the ethical tenets of research, human subjects protection, that are encoded in 45 CFR 46, and if you're doing FDA side work, 21 CFR 56. If you have institutional biosafety needs and reviews, if you're working with pathogens, toxins, recombinant material, radiation safety, if you're working with PET, other types of radiation, science review hopefully before you go to the IRB, have some form of peer review of your work. And if you have that type of regulated research, you're working on abbreviated IDEs, so regulated devices, or if you're working in IND side work, with drugs or biologics, small molecules, then you fall under provisions that are applicable to the FDA. So I have been really proud of the NIH because they actually had the first IRB in the country, which I didn't know until a couple years ago, formed in 1953. So this is a picture of the NIH back in 1953, and then a picture of the NIH today. And then you can find, these are the statutes that give you some of the responsibilities of an IRB. And it has different subparts if you were to look at it. This is one that this summarizes what we talked about in terms of all of the areas that really encompass a good clinical research program and support a clinical research team at the end of the day. And the whole job of this support is to take neurologic disease from bench to bedside and get it to our patients. This is work that I think if you would like it, I will share it, but there are algorithms to follow to know whether or not you're participating in human subjects research. It will walk you through, like this chart does. It can be very nice. My guidance to most people is if you are not sure, you can always submit to your IRB. Each IRB will have a slightly different variation on their thoughts in terms of the regulations. And when you don't know, it's never a bad thing to ask and get it in writing. There are algorithms for determining whether the NIH thinks something is a clinical trial. If you have a behavioral trial, another type of intervention, social behavioral intervention, maybe you're not sure, are you really using like a tool, or are you using a true intervention and changing something about what you're doing with the subject or a human research participant? And it will help you walk through different scenarios to determine whether or not you have a clinical trial and whether you might or might not be subject to CT.gov reporting if you fall under NIH policy. And CT.gov reporting is very important. It's a way in which we can disseminate clinical results. We can do it in a time-delineated fashion, and it has serious ramifications if you do not follow that policy. And it gives you some differences, right? So per FDA 801, an applicable clinical trial is normally phase 2. But with NIH policy, that can very well apply to most phase 1 work and BESH trials and other types of trials where you might not think that was a true intervention. So it's very helpful sometimes to walk through that algorithm, and the NIH provides that online for you. These are reporting requirements, some of the differences, and kind of the time delineation. You know, primary outcomes should be a year after collection, same with secondaries, and so those are going to end up reporting out differently. And this is clinicaltrials.gov and the failure to comply, which can be severe if you have grants. So these are some of the lessons learned I was talking about. We know that no man is an island in clinical research. You cannot do this by yourself. You try to do clinical research by yourself, you're going to rack up 1 million note to files and deviations very quickly. It's going to be impossible. So we tried as much as we could to utilize shared resourcing and to centralize resources as much as we could. And that was the walk through the clinical trials unit. That was really an exercise in amongst 21 and actually now 35 different PIs trying to centralize the resources we had in-house and then build them out to the needs of our investigators. And this was actually a slide I found from, I think, a capital financial company, but I thought it spoke a lot to clinical trials, except for maybe 5 acquiring complementary businesses. We don't acquire complementary businesses for the most part in clinical research, but we do identify shared resourcing and best practice, SOPs. We love to share those, CRFs for our databases, case report forms, trial metrics, auditing, compliance tracking. A lot of things published that can help us in our clinical research programs. Cross-functional collaborations are key, especially among different disciplines. Centralizing functions as much as possible to share the burdens of regulations and resourcing, especially in tough budgetary environments. Making sure we're compliant, you know, biorepositories, data repositories, those are all things that the current policies speak to, and sharing. And I changed 5 to be cross-collaboration with other researchers. And there's a lot of mechanisms out there where we could share. You can share with an intramural NIH. UL1s are a great example. Many shared research projects and collaborative trials, and most of our investigators love working with outside investigators, pharma, and other academicians. Site initiation visits. We have a large auditing group that started to implement CIVs very consistently a couple of years ago, so we did some work on it. And we looked at the reduction in noncompliance events on clinical protocols in clinical research once we had implemented CIVs. We found that at least in a subset of them, we could reduce noncompliance events by almost 50%. We thought that these types of meetings where you have you have your auditor, not normally your auditing team, but your auditor, sit down with the study team, walk through a very detailed checklist before study startup, after drug has been received, after they've gone through IRB, FDA, they've done their data management plan, they've created their CRFs, they have their initial regulatory binder set, right? All of those things have been done. You sit down with the study team, you walk through the checklist before they can start study. Make sure all the trainings are done, the nurses have been trained, and this has been very helpful. And it's helpful that even if the study team might not want to sit down with you for 2 hours to do this, or 3 hours, at the end of the day, it is a helpful exercise because people get on the same page, they ask their questions up front, and then everybody starts that clinical research program together. And that's what we have found, and we think it improves ultimate data quality and completeness. I think it helps to safeguard ethical principles of clinical research, and that it's valuable. Anticipating workload. This is one that we've only recently really been starting to sink our teeth into. And NHLBI, which is one of our colleague ICs at the NIH, has done this for a couple of years with their cell-based trials and their transplant trials. And they took an NCI metric called the OPAL, and they reframed it a little bit to try to score out protocols and the work of a clinical research team on that protocol to give it a complexity number. And then they use that complexity number to estimate the ultimate workload of the protocol. And it's quite a bit of work because the research coordinator each month will put in trial-specific data, and that research complexity score will change over time. It'll get an initial one, and then it will continue to update depending on how many people are enrolling. What's the burden on the team? How many visits are they doing? How many extra things are they having to do with the protocol? And that helps us resource allocate more effectively up front and also as the protocol goes. And so this is just the pyramid schema of kind of some broad-based things of what they collect with this OPAL score that speak to some central processes that are delineated, special procedures that are delineated all up front, and optional elements that are categorized up front. And these all go into the metric. And NHLBI has built it out actually in a Microsoft platform called Power BI. They have been willing to share it with everybody who's asked so far, so if you were interested, I would be happy to connect you. Data sharing plans and data management. This is a huge one for our teams lately with the new data sharing plan and data management requirements. So I just wanted to give you some helpful resourcing if you hadn't already seen it, and I suspect most of you had. In particular, that there is a sample data management and sharing plan online through the NIH. It can be, I would think it would be an immense challenge to craft one all by yourself with no template to guide you. At least the template can start to walk you through the thought process and lead you, other investigators, other support staff in the right direction according to the NIH policy. We have found, at least in our plans, that it's helpful, especially for our FDA-regulated research, and of course you must use a 21 CFR Part 11 compliant database in FDA-regulated research, but we try to use that as much as possible for all of our studies, especially if we have studies like a natural history study that will eventually bridge into an IND study, and then we keep everything consistent, including case report forms, use of CDEs, common data elements, and we try to, again, identify all the aspects of sharing and kind of keep it in a central location and have that clear plan as identified in that template plan that I shared with you. and then make sure folks have data repositories that they can access. We've actually started building one ourselves, but that's because we don't sit on the extramural side. There are many extramural data repositories available. And this is one that I just wanted to share here because I think sometimes it's under-recognized by investigators in terms of how you use adverse event toxicology criteria. It was my personal experience with one of my very experienced investigators, and actually I'm just going to jump, who was using the CTCAA version. This is an NCI toxicology criteria table that goes back to the 1980s. It's based off MEDRA terms. These are terms that come out of ICH. And it's a very widely used table that grades out adverse events in clinical research and has them by organ system class that we use in our trials and NCI uses in their trials. And it's probably one of the more robust ones. I listed out the ones that I am aware of. If you have ones to add, please tell me because they're hard to find. The most common ones are CTCA and DAIDS, and FDA has their own that really speak to healthy volunteers in vaccine trials. And then there's a rheumatology one, which I have never used, which is the RCTC. But I realized that some of my investigators were not understanding the flow of how to use the AE tables, and I oversee our DSMB, our central DSMB, and I realized this and I think that was my beep, when I was looking at their AE logs. And when you look at these tables, you go to the term, you go to your system organ class, you try to identify the best term, and then you grade out the term. And you try to be as consistent as possible so out the lifetime of your trial, and in trials that are similar, you can be consistent with your terms across trials so that you can understand the data across trials better because you're using the same language for the same events. And you try to get the most comprehensive term for whatever it is that you're seeing on a patient. And I just copied, in the nervous system disorders here, myasthenia gravis, grade it 1 through 5, and you can kind of see how CTCA will break that down. Whereas if you were going to grade out neuralgia, you think neuralgia is the best term, you can't even go past grade 3. So this table has very defined ways in which it wants you to grade things, and then you can be consistent. Or if you're in a group, some groups choose to use grade 1 through 5 for their adverse event grading, some choose to use mild, moderate, severe. Right, but just to be consistent about the framework of your AE grading. So we found that to be very helpful, and to walk our investigators through how you navigate whatever criteria you're using. You're using CTCA, this is kind of how you navigate that table. It wasn't something we realized up front, but it was definitely an issue, and it was very easy to fix. So clinical research team, it's a team sport, and we love the intramural program, and we're glad we have our field of dreams. And these are some training opportunities that I'm aware of. I have taken or have participated in the first 2 on this list. I have never done the third. It's a basic neurotherapeutics course. The Ascent course is more clinical. The R25 used to be called the Vail course back in the day. That's the CTMC course run out of extramural NINDS and normally the University of Michigan. And then the FDA puts on an investigator training course every year. Sometimes it's in person, sometimes it's online. I'm not sure if they're having one this year. It was a little spotty during COVID, but when it does occur, it's very good. So I would recommend looking for it. Duke has a variety of different clinical research training programs, including a master's program. I have done those didactics, they're lovely. If you wanted kind of a full didactic program, that's a good one. And I've actually always enjoyed the Row webinars, so I put them on here. That's a CRO. I received no money from any of this. I'm just a federal employee with no other funding sources. But they do have some nice, I think, basic topics that cover the gamut of clinical research, and they're available for free online, so it's a bit interesting. NIN's intramural program puts on biostatistics lecture series everywhere, and we videocast them, so that's that. And then NeuroNext has a really nice variety of SOPs and other resourcing for trials teams that are available on their website, and so I put that link there too. Thank you very much.
Does anyone have any questions? That was brilliant, Rioma. Just those training opportunities for the European and the UK early career researchers, can we access those from abroad? Or no?
I don't know about the CTMC, but the CTMC, even if you couldn't get into it as a participant, they have all their lectures online, going back many years, all the way to Vail. So that's wonderful. All of our side resourcing, you could. They're certainly available online. And I think Ascent would not be a problem, and I don't know about the other R25 program.
Are there any other questions from the audience?
Brilliant.
Thank you so much.
So we're going to transition over, and Dr. Rioma just gave a great talk, and the end of the talk was about resources. So we're going to spend a few minutes talking about resources that the NMSG can provide to both more junior investigators and more senior investigators. And also we'll call your attention to the gentleman in the back of the room on the left there. There are some signature cocktails available if you want to take advantage of those during the session as well. We're going to have, Dr. Wenzel is going to talk about some of the ways that you can partner with NMSG to do research.
Thank you. I'm not a physician, but I appreciate the the promotion. My name's Marie. So we just wanted to share some of our NMSG resources that we have available to all of our NMSG members. So if you see this link here that's highlighted, there's a short application that you can access on our website. And this application is for grant support or project support, and they include this menu. So from things simple to complex. And these are resources that some we provide for free, or some you can build into your grant application and incorporate into the funding that you receive. But it goes from brainstorming. If you have an idea, you can run it by the executive committee and get support on that. Building a budget, assembling a grant. Obviously, there's more to putting together a grant than just the science. So we have experts that can help you piece together all the grant documents, or find sites and piece together the site documents. Statistical consults, and then support during the course of your study. Database management. Obviously, we have a great data manager and database team at University of Rochester. That infrastructure already exists, so you can tap into that. Protocol building, or even just instructions. So this is open to any member of NMSG, from senior to entry-level investigator, you name it. So we just want to make sure everybody knows about this. We're looking for applications, and please share with your colleagues. The executive committee meets quarterly, and one other thing, even if you'd like them just to review your grant application, you can list in your grant application that you went through an NMSG review and got an NMSG endorsement, which could be a nice little plus on your grant application. We also wanted to share the Shark Tank, which is for early investigators, early career investigators. So this is a nice little seed funding. We will see our Shark Tank applicants on Sunday at this meeting, but we do this annually, so people can apply to present their project ideas to the sharks, who are experts in the field, and get awarded. One person is selected, I think once we had two, right? And get a nice little seed funding. So then that will make the project automatically become an NMSG study. And then we've even had these awardees come back and present at the next meeting, and so it's just nice support from your colleagues to share the update of your progress of your study. Also, even if you don't win Shark Tank, you've still presented your idea to experts in the field who may help with future collaborations, or so on, or good practice just to put together a nice elevator pitch and really hone in on your ideas for a project. So then I think then we'll discuss the fellowship opportunity. Thank you.
So I just wanted to, I'm going to pull up slides in a second. So I just want to call people's attention to an opportunity that can support junior investigators getting their research project off the ground. The NMSG co-sponsors with the American Brain Foundation, the American Academy of Neurology, a clinical research training scholarship. And those are the two appropriate websites that you can look at. These can be game-changing grants for someone getting a career off the ground in clinical or translational research. So the NMSG award is $65,000 a year in salary support plus $10,000 a year in research support. You can get some additional support outside as long as it doesn't exceed the amount of the NMSG award. And it's for 2 years, and it's available to anybody who's an AAN member. It has to be somehow patient or translational research. You have to be within 5 years of residency graduation. So fellowship does not count. The clock starts when you finish residency. And if you get our award, we require you to come to the meeting and present your research. Tomorrow night, we're going to be having our current fellow present at the keynote session. Why do an NMSG fellowship? It provides you with protected time where you can complete a research project. It also requires that you complete a research training program. The program that was just mentioned, the Clinical Trials Methodology course that's run through the University of Michigan, is an excellent thing that can be included in part of this program. It allows for mentorship, and it allows for a lot of networking at meetings like this one, and also through the American Academy of Neurology. There's a special mentorship and education program that comes along with winning one of these awards. So if anybody's interested in this, this is what you should be thinking. So you need to think about the project early. It's a fall deadline, so you would need to start thinking about your project now for a submission next year. Identify your mentors early. Identify education resources to include as part of your application proposal. And then you need to talk to your chair, your division head, or your program director as soon as possible if you're thinking about one of these. These are not enough to cover faculty salaries, but there are many chairs who are supportive who will allow you to cover your salary with a program like this because it's an investment in a junior faculty person. So it's something that you can look into, and it's something that changed my career getting one of these awards because I had a supportive chair. So these are some of our NMSG fellows. I know at least 4 of them are here today. Dr. Katz is going to be speaking tomorrow night. Dr. Rabin, I saw walking around earlier. Dr. Gonzalez-Perez is here and is going to be part of the session in a few minutes. And Dr. Sayed Sajjadi is also here at the meeting, is going to be speaking. And just so you know, there are other versions of this type of grant mechanism that are related to the people in this room. So there's a training scholarship in ALS available through the same program. There's one for peripheral neuropathy through the same program. There used to be one through Myasthenia Foundation. I don't know if it's just not offered this year and it'll be offered again next year, but that's another thing to look out for. This is all on the AAN website. And you can be an international applicant. You just have to be a member of the AAN. And this is another award. This is a more general career development award through the same program. It's a bit bigger, it comes with a bit more money, a bit more competitive than the disease-specific ones, but it's something to take a look at. Thank you.
Okay. So the next section is, we have an icebreaker and then breakout groups. So the more senior mentors or investigators are welcome to stay because, we'd be grateful for your imparted knowledge as well. Or, you're welcome to leave as well. So I'm going to hand over now to Dr. Karen Sutherland, who's going to do our icebreaker session. Thanks, Karen.